All right, I got the thumbs up. All right, well, welcome everyone to the 2024 Southern Flounder Symposium. I'm Steve Poland. I'm the Fishery Management Section Chief at the Division of Marine Fisheries, and I'm really excited to see the turnout that we have, and I appreciate everyone that took the time out of their day to make it out here. So before we get started, I just wanted to cover a few housekeeping things. So there are complimentary beverages at the back. There's coffee, tea, and water, so please get up, get yourself a drink. I will ask if you need to leave the room, maybe to go to the bathroom or go out and take a call. Try to do that discreetly, and if at all possible, try to wait in between presentations, just not to throw off the flow of the presenters. And then lastly, if there is time for questions after presentations, we'll facilitate that. We have microphones set up throughout the room, and we've got staff stationed at each table. So if you have a question, just raise your hand after the talk, and we'll have a staff member run over to you with a microphone. So with that, I'd like to invite Director Kathy Rawls up here for some opening remarks and to kick us off.
Thank you, Steve. Good morning, everyone. I think I'm going to help Steve and his team out this morning right out of the gate. People came here for the flounder. I'm not the flounder, so I'm not going to take much of your time. But I did want to, on behalf of the Division of Marine Fisheries, welcome everyone to the 2024 North Carolina Southern Flounder Symposium. As Steve said, my name is Kathy Rawls. I am the director at the North Carolina Division of Marine Fisheries. I've been with the division for 28 years, and May of this year will be my third year as director. I started my career in 1990 with the Division of Marine Fisheries as a river herring technician. I worked out of the Elizabeth City office. I've worked my way up through the division, district biologist, district manager, section chief for fisheries management, and on to where I will end my career, hopefully, as the division director. And I say all of that just to really say that you are surrounded by people today whose stories are very similar to mine. There are people here who have dedicated their careers to fisheries, data, science, management, and I can assure you that you don't do this for the fame and fortune. That's not why we're here. We do it because we love this stuff, and we are super excited to be here today. And really, this is an exciting day for anybody that cares about flounder and knows anything about flounder and is interested in the species. It is especially exciting for this division and this staff that has really poured their heart and soul into putting this symposium together. Thank you all so much. I know what this takes. I have seen the work. I've watched you work, and it's great. Everything looks great, and I know that this is going to be a great day for all. I personally have been looking forward to this day for a long time, and so have many other of our staff members. So I'm glad it's here, and we are very excited, as Steve said, about this opportunity. Thank you as well to the researchers who are here with us today and have agreed to share their knowledge, their research, and their findings with us. It's a great opportunity. And certainly, last but not least, thank you to the commercial and recreational fishermen, the consumers, the fishermen, the stakeholders that have joined us here today. We appreciate you taking the time out of your day to come and learn more about southern flounder here in North Carolina. I know the staff is probably tired of hearing me talk about this, but I talk about it everywhere I go. One of the goals that I have had as the division director was to increase messaging and outreach and education in this division. We can have the best fishery science and data in the world, and if we can't message and communicate to our stakeholders what it means, how is it collected, how do we analyze it, how do we use it, it will not be effective as it could be if we had more improved communication, education, understanding, collaboration, the type of collaboration that we are doing here today, buy-in, and really trust in that we are all in this together. No matter which side of the ball we come from, we are all doing this together. This is an important piece of the messaging in my mind. Staff had a lot of discussions internally about what they wanted to get from this symposium and really what we wanted for you all to get from this symposium. And it's meant to bring researchers and the public together with managers to discuss this topic of interest, in this case, everyone's favorite southern flounder. It is a very important commercial and recreational fishery to our state. It's very important to our fishermen and our consumers. We are really excited that all of these groups are represented here today, and I am confident that all of the recreational, all of the commercial fishermen, all the managers, all the researchers, everybody that here is going to leave this symposium with a little bit more knowledge about southern flounder, and I think that is the point, and have met a lot of different people. So I'm looking forward to that. I want to impress upon everyone that's here today that this is a neutral setting. This is relaxing. We are all here to learn about southern flounder research that is occurring in North Carolina, and about what we know, maybe more importantly, what we don't know. What are the questions that we still need to answer? And where are we trying to go, and how exactly are we going to get there? These are the things that we want to talk about and that we want you to learn about today. Everyone here presenting, whether it's a talk or a poster, is helping to add to the knowledge base that we have about southern flounder and to help ensure that we are using the best data possible, which leads us to the best management possible. During today's symposium, we have talks about life history, habitat use, movement and migration, and the data that goes into stock assessments. There are posters on additional university research. There's also some posters outside on DMF data collection and how it's used. There's folks manning these booths all across the venue. So it's a great opportunity. Marine Patrol is also here to answer any questions that people may have about enforcement. Additionally, we want to acknowledge and thank our keynote speaker, John Carmichael. John, where is John? He's got to be here somewhere. There he is. Yep. John is the Executive Director of the South Atlantic Fishery Management Council and formerly a stock assessment scientist for the Division of Marine Fisheries. Now, just a little story about John and one of my first interactions with John. I was a new biologist in Elizabeth City working on river heron back in John's days as the DMF stock assessment scientist. And I remember that he graciously agreed to review one of the first technical reports that I had written outside of the college setting. And now, mind you, my mother was a school teacher, and when I was a young child, I used to help her grade her papers. So I realized right away that all that red ink was not good news for me and my paper. But I still have that paper in my desk. I'm not kidding. And it was a little traumatic, but I learned from that, and I carry a lot of that with me. The attention to detail has got to be something that I've carried through my entire career, and spelling is very important. And thank God we have spell check now, John, I've always remembered that, and I appreciate that. And I will also tell you that John is a wizard at breaking down technical stock assessments and the details for better understanding. So if you've got any related questions like that while you're here, don't hesitate to hit John up. He's got a lot of experience. His talk this afternoon should tie together the subjects of today's presentations, and it'll help address some common questions and concerns about fisheries management and science, and considerations and lessons learned from a long, long career in fisheries. John has a lot of experience to pull from, and we are looking forward to his presentation this afternoon to help us tie all of this together. I want to impress upon everyone, again, how lucky we are to have this opportunity to have all of these researchers, all of these biologists and managers and stakeholders that use the resource in the same room today, all day long. This is a great opportunity. We encourage you to mingle and talk to folks that you've never met and people that you have. Talk to the staff, talk to the researchers. As I said earlier, we love this stuff. So this is what we live for, and we really are looking forward to this. You can also learn how you yourselves can get involved and help answer some of these research questions through citizen science. Talk to the staff about these things. Engage with your fellow citizens and your stakeholders, and find out what people are interested about this species. What are the things that concern other people? What are the things that attract them to flounder? What do they do? Do they just eat flounder? Do they fish for them? I think you'll be surprised across the board how much common ground we actually share when it comes to fisheries in general, but certainly flounder is what we're focusing on today. And I just wanted to make a quick note to highlight that what today is not. So today, thank the Lord, this is not a management meeting. We are not making management decisions. We are not making any decisions. The worst thing you're going to have to decide today is where you're going to eat lunch. There are no action items. There is nothing like that for us to do today. All we have to do is learn. This is a rare opportunity, as I said, for all of us to learn about this topic. It's a shared topic of interest, and about this ongoing research, which is always interesting. And we ask that throughout the presentations, please be respectful to the presenters and to each other, and help us make the most of this opportunity and the time that we have invested and that y'all have invested to be here today. It doesn't matter if you are commercial or recreational, if you're a consumer, a manager, if you're just interested, or even if you're here by accident. It doesn't matter. We are here to learn from each other, to learn about this species, and this is a great opportunity for us to do exactly that. The Division of Marine Fisheries is dedicated to ensuring sustainable marine and estuarine fisheries and habitats for the benefit and health of the people of North Carolina. This is our mission statement, word for word. But we cannot do that without people just like y'all. Better science leads to better management. And this type of learning and this type of collaboration, just like we're going to do here today, is exactly what leads to better science and ultimately, I believe, what will lead us to better management. We can't thank you enough for your attendance. We can't say that enough, how much we appreciate you taking your time and participating in this unique event, highlighting Southern Flounder in the great state of North Carolina. What better way to spend your day? We welcome you, we thank you, and we hope everyone enjoys the symposium.
All right, thank you, Director Rawls, for setting the stage for the day. So we're going to ask our first speaker to make his way up here, and I've got the pleasure of introducing Dr. Fred Scharf. He's a professor of biology for the Department of Biology and Marine Biology at UNCW, and I believe recent department chair, right? Not yet, okay. We'll hold off on that. But Fred possesses a PhD in wildlife and fisheries conservation from the University of Massachusetts in Amherst, and his research at UNCW is focused on population dynamics of marine and estuarine fishes, including the study of recruitment processes, the estimation of mortality rates, fish reproductive dynamics, and behavioral ecology, particularly movement and migration. And you left off offshore pelagic food web ecology. That was a big one you did recently. So. But Fred, he supervised a thesis and dissertation of more than 20 UNCW graduate students, and many of those students work at the division or have come through the division. So won't take any more of your time. We've got Dr. Fred Scharf. He's giving us a talk on the key aspects of southern flounder life history, identifying knowledge gaps and potential effects on stock dynamics.
Well, thank you, Steve. I appreciate the invitation to be here, and it's nice to see so many folks this morning that I've known for many years, and folks that I've just met recently in the past few years. And so, as Steve said, I've been at UNCW since 2002 and have been engaged with fisheries management here at the state level since I've started. and over the last 7 or 8 years have become more involved in federal fisheries management as well. So, and the work that we've done on Southern Flounder started way back in 2005, and we've been studying aspects of Southern Flounder life history and ecology ever since. And my goal for today was to provide you with an overview of the life history of Southern Flounder and talk with you about some of the knowledge gaps that remain, talk to you about a summary of some of the research, not only research that we've done, but research that colleagues have done, both in this region and throughout the range of the species, to inform what we know about their life history and how these key parts of their life history affect how we manage them, and how we assess them as a stock. So before I get started, I just want to acknowledge the many students and collaborators that have contributed to work in my lab, as well as projects that we've done in the region. And so you see pictures up there of several graduate students. You don't see Steve. Steve was a student in my laboratory, but Steve worked on offshore pelagic fishes, as he mentioned. But lots of undergraduate students, technicians, as well as collaborators both in North Carolina and throughout the South Atlantic and Gulf of Mexico. So I want to acknowledge all the contributions of those folks. Okay. So as many of you know, the Southern Flounder has a distributional range that occurs throughout most of the South Atlantic. North Carolina has long represented the upper part of the range along the South Atlantic coast. They continue down to the central part of the Florida coast. You'll notice a gap around the bottom of the Florida Peninsula, and then the species continues throughout the northern Gulf of Mexico. And aspects of the life history that I wanted to talk about include several, and I'm going to try to use this pointer without leaving the mic too much. But in terms of the life history of the flounder, we start out here with oceanic spawning. One of the first things I'm going to talk about is what we know or don't know about these ocean spawning locations. But the spawning takes place in the ocean, after which there's a fairly lengthy larval period that can last anywhere from 30 to 60 days. And so when the larvae show up at our coastline and enter our estuaries, they're already 1 to 2 months of age. And then they are advected into our coastal estuaries and river systems, where they settle and spend the first generally 2 years or so of their life growing in our estuaries, at which point they reach sexual maturity, emigrate to the offshore environment, and participate in spawning. And so I'm going to talk about several aspects of the life history in terms of what we've learned from recent research over the last decade or two about some of the ocean spawning locations, about processes that affect this larval transport, settlement habitats, and how those settlement habitats and those processes post-settlement influence or can potentially influence recruitment. Talk about movement and migration out of the estuaries, which you'll also hear about in some other talks today, and eventually reproductive success and recruitment success of adults, which is a pressing issue. So I'll start with a little bit about what we know about offshore spawning locations. And up until recently, most of what we knew was informed by fairly old surveys of egg and larval stages, and these list several of those surveys. None of these surveys were designed explicitly to capture southern flounder, but did capture southern flounder during the winter along most parts of the South Atlantic coast at one time or another. So the first surveys happened in the 60s and off the shelf in December, others in the early 70s and late 70s, and then there was a SABRE project, a South Atlantic bite recruitment project focused mostly on menhaden, but it also captured southern flounder larvae in the winter. And the the take-home collectively from all of these resources was that most of the larval flounder were captured over deeper waters in the outer part of the continental shelf. So catches, all the stations in this survey, for instance, that had large catches occurred at waters greater than 40 meters in depth. Another set of studies that informed what we thought about spawning locations in the winter were two fairly comprehensive diving surveys that took place in the past 2 decades. So one about 20 years ago off the North Carolina shelf that was conducted by the Division of Marine Fisheries, and it was conducted mostly at artificial reef sites as well as some natural reef sites, all at depths generally below about 130 feet or roughly 40 meters or so. And one of the things that they observed was that they saw and encountered Paralichthys lethostigma in shoal habitats during the fall, but in the winter, only 7 southern flounder were observed in the winter during those surveys at those depths. And similarly in South Carolina, in the latter part of the 2010s, they did a fairly extensive survey off the South Carolina coast. Again, most of the dives were at scuba depth below 40 meters. No lethostigma were captured despite 91 winter dives. Lots of Gulf flounder, lots of albigutta, which is a sister species to southern flounder, but no lethostigma. Again, pointing to the idea that the lethostigma during the winter were deeper than 40 meters. And as I said, you'll hear more about this in a later talk that Shelby's going to give that includes some information that we've learned from 2 different types of satellite tags. And there's a poster out in the lobby that includes information from some recent satellite tag data that we've obtained. And the take-home is that these offshore spawning areas still remain a bit uncertain despite some of the new tagging data. And so some of the conventional tagging data that I'll summarize for you in a little bit pointed us in the direction of some other hypotheses. And so far, the satellite tagging data does not align with some of those hypotheses. So I know Shelby will talk a little bit more about that. And you can look at the poster in the hallway as well. After spawning, the larvae, again, have a 30 to 60 day larval duration before they're transported into the estuaries, where they undergo metamorphosis into a flatfish and begin to grow and feed as an estuarine-dependent species. And so some work that was done by Chris Taylor, who's at the NOAA lab in Beaufort, started to look at environmental conditions and nearshore ocean conditions that might affect the transport of larvae into the estuaries and coastal rivers. And what he found was that winter winds, specifically winter winds that had north and east components, right, showed these positive relationships with the juvenile abundance index in the estuarine systems. And so one of the surveys that's carried out by the North Carolina Division of Marine Fisheries is an extensive trawl survey that's done in the spring that captures young of the year southern flounder that are anywhere from 4 to 6 months of age. And he found that that index was really strongly related to some of those wind conditions. The larval signal that they were seeing at the coast also was retained. If you saw good larval signal in the winter, you saw good recruitment in the spring during that May-June trawl survey that captured young juveniles. So if we saw good recruitment coming into the inlets when the fish were 30 to 60 days old, we tended to see that signal maintain itself at about 4 to 6 months of age. Some of the things that we've focused on more recently, both in our region as well as throughout the range of the species, have to do with juvenile settlement and habitat effects on growth and contingent formation in southern flounder. And so the long-held view is that one of the things that's really unique about southern flounder life history is that the settlement has occurred by and large, in lower salinity habitats. And so particularly along the South Atlantic coast, we see the bulk of the settlement, the higher densities of the settled fish, occur in these low salinity habitats. We do see settlement in mesohaline conditions in mid-salinity areas of the estuary, but it still has been more dense in the low salinity areas. And the long-held view is that this happens in the late winter, early spring, peaks February, March, and April of settlement. And then in the summer, we start to see the flounder slide down from those oligohaline or low salinity habitats into the mesohaline. But some of the recent work, both in the Gulf of Mexico and here in North Carolina, has suggested the potential for low salinity contingents. So fish that don't move out of those low salinity habitats and remain in higher salinity waters or low salinity waters throughout the estuarine period. This was a recent paper that was published in the Gulf of Mexico, some work in Mobile Bay, that just shows the probability of being an estuarine resident or a freshwater resident, depending on where they were caught, and also depending on discharge, so flow rates and precipitation rates during different years. And so these are fish in the red and green lines that were captured up in the delta or up in the upper headwaters of the Mobile Bay estuary, and their probability of being freshwater residents based on the chemistry in their otoliths was affected by where they were captured as well as discharge. And the other thing about these patterns is that the level at which these freshwater fish are then able to contribute to the fishery in the future is uncertain. They found in Mobile Bay that they didn't contribute much. And so most of the landings that were happening for 1 and 2 year old flounder did not include many of these freshwater contingent members. Some work that we've recently done here in North Carolina, some work that was done in the Cape Fear River, was looking at the effects of these habitat settlement choices and whether flounder are settled in these low salinity areas versus more moderate salinity areas, and how it affects early growth and mortality patterns and the formation of those contingents, ultimately recruitment success. And one of the things that we see is when we look at the sizes of fish over time, which is here, in the purple are fish that are settled in those oligohaline habitats and remain in those habitats. And in the blue or greenish color are fish that have settled in the mesohaline or have shifted into those habitats post-settlement. And we see much greater opportunities for large size and growth in those mesohaline habitats. And we hypothesized that this might be due to some osmoregulatory costs of being in those low salinity habitats. But what we found, by and large, that it wasn't physiological, but it was mostly food related and the availability of certain prey that delayed their shift to feeding on fish. And so when young flounder begin to feed on fish, their growth rate accelerates tremendously. And this result is aligned with some results that were seen by some folks at NC State that were studying these same phenomenon in the Neuse River 20 or 30 years ago. And so the take home is that what we've learned is that some of the juvenile recruitment patterns can be linked to local environmental conditions, particularly wind patterns in the coastal ocean at critical times of year that affect their transport. We still have a fairly poor understanding of the conditions in the coastal ocean necessary for successful egg hatching and larval survival. I'll touch on that again at the end. But one of the things that we still are looking to study are some of the implications of different post-settlement contingents in freshwater and mesohaline habitats and how they contribute overall to the dynamics of the stock. Another key part of flounder population dynamics is our key life history traits, one being the maturity schedule, the timing of maturation within the life history, other traits being growth rates throughout the life history. One of the things that we did during 2009 and 10 was to start to study the reproductive patterns of southern flounder in North Carolina and some of the other states to the south. And by using histological analysis of gonadal tissue, of ovarian tissue for females, we were able to determine that the timing of maturity within the life history was happening later than we had presumed, at least here in North Carolina. And this had some important implications for, let's see if I can get this to work, had some important implications for the fraction of the harvest that was mature, right, being much lower. Only about 45% of the fish at age 1 were mature, as opposed to previously we had assumed it was closer to 75 or 80%. It also had some important implications for the harvest rates that the stock could withstand and still contribute to a specific threshold of biomass that we thought was necessary in order to preserve the stock. So it had some important implications. What we've seen in other regions since then is that we think that the maturity schedules may display a latitudinal gradient, in that the pattern that we see in North Carolina here is not universal for the stock. We saw anecdotally some very interesting patterns off the coast of Florida, off the east coast of Florida, that suggested that maturity wasn't size dependent within the ranges that we were looking, but had a really strong seasonal component once the fish reached a certain size. There's also been some new data from the coast of Mississippi that suggests in those warmer water areas, the southern part of the range, that the flounder mature even earlier. Some recent work on growth variation across the range has also demonstrated that there's lots of opportunities for growth to vary at scales of estuaries and coastlines. And the processes that drive that variation are still a matter of study that we don't understand. And so those are areas for future study as well. Particularly, North Carolina took the lead on developing the first coast-wide assessment for the South Atlantic. The Gulf of Mexico is starting to go in that direction as well and assess their stock from Texas across to Florida as well, as opposed to independent units. And having a really good understanding about how the life history traits, these key ones, timing of maturity and growth, how they scale across those ranges is critical. One of the funnest projects that we did started back in 2012, and Steve was in my lab at that time and got to participate in some of this work. But we were asking a few questions about movement and habitat use within the estuary during the time when flounder are residing in estuaries. We also wanted to look at the timing and pathways of their migration out of the estuaries. And we also wanted to look at the fates of fish being exposed to different sources of harvest, both commercial and recreational within the estuary, as well as sources of natural mortality. And we did this through a combination of conventional tagging at a very broad scale across the state, but then a very focused acoustic tagging or electronic tagging study. And this was conducted in the New River, about an hour south of here, off near Jacksonville. And we were able to use multiple acoustic listening stations, which are indicated here in black, to track the movements of fish over long periods of time, up to 2 to 3 years, that they were able to maintain these tags. There we go. Okay. One of the other things we did during this study is we were able to do some really fine scale work in one of the tributaries, Northeast Creek of the New River, to look at associations between flounder and certain features of the habitat that might contribute to their distribution. So one of the things that we found in that system, we were tracking fish on daily basis, weekly basis, and we were able to identify some consistent areas of aggregation, and that the flounder during long periods of time displayed very low activity, mostly from late spring through early fall. So for 4 to 5 months, right, we had fish that would stay within areas that were only 1 kilometer squared and would stay there for 3 to 4 months consecutively without leaving those areas. We found that these areas were also associated with high prey fish, both high prey fish diversity and high prey fish catch rates. So we looked at the things that the flounder were eating and then looked at their distribution in that system and found that the flounder were aggregating in the same places where those species aggregated. In terms of the migration events, the New River was a really nice system to work in because it had some really nice natural migration corridors, which you can see here in different parts of the river where there are some natural pinch points. We could build in some redundancy with the listening stations and detect fish moving through the river and look at the timing of their departure and the pathways that they used. And one of the things that we found is that in the New River, the emigration timing was really consistent and predictable. About 80% of the fish left between the middle of October and the middle of November, with about 15% of the fish or so being late and only about 5% of the fish being earlier than that window. The other thing that we found was that the migration happens really quickly. So when the fish initiate migration behavior and start downriver movement, they emigrate within 7 to 10 days. So they don't spend a lot of time stopping. This is the emigration timing across that system, and so you can see the window between mid-October and mid-November, and this was over 5 consecutive years. From 2012 to 16, we monitored emigration of these tagged fish for 5 years in a row. And within that window, you see a little bit of year-to-year variation. The peak window is still the same, but within that window, we had 1 year where a lot of fish left very early in the window and another year where they left late. And those were tied to environmental events, particularly the cumulative impacts of precipitation, wind, drops in barometric pressure as we get fronts moving through the estuary. It was interesting to us that we didn't see them respond rapidly to a single front, but it was more consecutive fronts that we would see that would initiate movement of fish out of the system. And I know that Dr. Ash is going to talk a little bit more in her talk about emigration patterns that they saw from Pamlico Sound, so a different system within the state. The other thing that this acoustic array allowed us to do was to look at fates of fish. So fish that were harvested recreationally or commercially, fish that died naturally, as well as fish that emigrated. And we combined that with the statewide traditional tagging of fish to be able to estimate the fates of fish at different scales. And what we were able to do with using a system of double tags and high reward tags is we were able to estimate separately rates of harvest from commercial fishing and recreational fishing, natural death rates, and emigration. And what we found, which was really interesting, was that the models that were most supported had consistent natural mortality rates from year to year, which is in the green here, both at the scale of the state, where it says North Carolina, and at the scale of the New River. The same with emigration. What's interesting is that we found that emigration from the system made up a large fraction of loss. And only through the use of the electronic tagging approach could we separate that from natural death. And then the fishing mortality varied here in red, both at the North Carolina scale as well as at the scale of the New River from year to year, which we'd expect based on certain conditions. So the combination of habitat use and emigration and fates of fish allowed us to make some general conclusions about the fact that we see limited estuarine movements for a long period of time compared with really rapid movement and extensive migration that was consistent in time. The emigration accounted for a very large fraction of total loss, but we still don't have a good understanding of the sources and the size dependence of natural mortality. We were able to estimate natural mortality, but not identify the sources. One of the last things I want to talk with you about is the scale and extent of stock mixing in the ocean. So when these fish migrate, one of the things that we've seen from conventional tagging that goes back into the 80s, and this is conventional tagging that we did, North Carolina Division of Marine Fisheries staff did years ago, staff in South Carolina and Georgia did as well, is that we see extensive movements in a southerly direction. So any flounder that moved more than 50 kilometers was always recaptured to the south of its release location. And you can see the extent of those southern movements all the way down to Florida. And so it got us thinking about degrees of exchange among different states. And so we looked at a couple different ways to look at mixing. One was looking at morphometrics, and the morphometrics told us that we had extensive mixing in the South Atlantic. We couldn't reclassify fish to state based just on morphometrics alone. So there was lots of mixing. We also did genetic work, compared flounder from different states in the South Atlantic as well as the Gulf of Mexico. We see a clear split between the basins, between the Gulf and South Atlantic, but we see limited structure within the South Atlantic, and most of it's not aligned geographically. So we may see some variation specific to different systems, but it wasn't aligned geographically. And in the marine communities, we assume most of the time that this mixing is generated mostly during the larval transport stage. Recall, flounder have 30 to 60 days in the offshore environment. One of the ways that we looked at that was to look at the chemistry of the otoliths, the ear bones of the fish, and we first did that by collecting young fish that hadn't left the estuary, and we looked at their juvenile signatures. And then we recaptured fish when they were 3 or 4 years old and looked at the chemistry of those same regions. And what we found is that fish that were captured in North Carolina and South Carolina spent their juvenile time in any number of the states in the South. So you can see here North Carolina fish came from all 3 states. We didn't have good data from Georgia for this study, but we had good data from those states. And the same with fish from South Carolina. And so what it suggested to us was that the mixing, genetically and morphometrically, was at least partially driven by movements of post-spawning adults. Okay. So lots of mixing, post-spawning adults, and ocean estuarine habitat use is still uncertain. So to summarize these outstanding questions that remain, right, is still offshore spawning locations, conditions that support larval success and transport, some of the impacts of where estuarine settlement occurs and the formation of contingents, how they contribute to recruitment, the spatial scaling of life history traits, whether that occurs at a very broad scale or whether there's really fine scale differences, the dynamics of natural mortality. We've been able to estimate it but not understand the sources of it, and then understanding the effects of stock mixing and how much post-spawning adult movement affects that. I'll leave you with just a little bit of a downer story on sort of recruitment of southern flounder. This is sort of the most pressing issue. When we had a workshop in Baton Rouge, Louisiana 2 years ago, we spent a lot of time with folks in the Gulf and the South Atlantic talking about this pattern. This is a pattern of abundance of adult southern flounder along the Texas coast, and you can see a pattern of slow and steady decline since the 1980s. A more recent paper that looked at juvenile southern flounder abundance across different bay systems and rivers in the Gulf of Mexico saw similar patterns, where you see the states, that the systems that I just highlighted in red have showed long-term declines that date back into the 80s. And you also see systems that have more recent declines that started in the early part of the 2000s. And when you look at in the South Atlantic, we see similar patterns. We see some areas, some systems that show longer-term declines in juvenile recruitment, and we see other systems that show more recent declines that started more in the last 2 decades or so. The bottom line is that we see range-wide declines in recruitment, North Carolina to Texas, that we don't fully understand. And one of the things that we've talked about, and what I'll leave you with here, is what we're thinking about are impacts of warming ocean conditions. Southern flounder are winter spawners. The climate literature has shown us that the impacts of climate are most extreme in the winter. The winters just aren't getting as cold anymore. And so the temperatures that southern flounder need for successful egg hatching and larval growth and survival are really uncertain. We know somewhat about what we're able to do in the hatcheries, but we don't know if they're able to find those conditions in the ocean. And so we already know that the temperatures in our estuaries are affecting sex ratios of southern flounder, but we don't know about offshore conditions and whether they'll support continued recruitment success, and how quickly the southern flounder will be able to adapt to a warmer coastal ocean. And with that, I'm happy to take any questions.
All right, thank you, Fred. We've got time for one question. Took most of your time.
I know.
That's good, a lot of information. And if you don't have a question yet, I mean, Fred will stick around all day, certainly go up and talk to him. Yeah, right here.
Okay, so with the trend that you're seeing on decline in recruitment, are we certain that this is an environmental factor? Is that the assumption here? There's something going on out there?
Well, no. I mean, we're not certain what it is, but the fact that we're seeing it across the range in places where both fishing pressure and sort of management intensity vary quite a bit, right? So in places where the harvest pressure has been both high and low, places where management intensity has been high and low, we're seeing the same patterns across the range. And so one of the things we've seen in other aspects of southern flounder life history is that we do see synchrony at fairly large spatial scales. In other words, we see flounder that occupy a fairly broad range will respond to environmental conditions the same way. So we're looking at this recruitment question the same way. It's troubling because we're seeing the same challenges with other fishes. Some of our federally managed fishes in the South Atlantic are recruitment challenged, despite lots of management intervention that has rebuilt biomass of adults. We're still seeing very low recruitment in many of those stocks that we don't understand. And so we're starting to look experimentally at some of these things with southern flounder in terms of how sensitive they are to winter temperatures. And so we think that there may be an answer in there, but we've only just started to touch the tip of the iceberg.
All right, thanks, Fred.
Sure, thanks.
All right. Very informative talk. We appreciate that. So next, while he makes his way up, I'll introduce Dr. Martin Posey. He's a professor of biology and marine biology at the University of North Carolina at Wilmington. He runs the benthic ecology lab there at UNCW, and I think there's a few alumni from that lab that work with us at the division, including one of our symposium planners, and Mark within the back. Martin holds a Bachelor's of Art in Zoology from UNC and a PhD in Biology from the University of Oregon. His projects and research interests include oyster reef restoration and monitoring with a focus on fishery impacts and habitat mitigation. And he was also a former member of the Marine Fisheries Commission. And you enjoyed all your time there, didn't you? So today, Dr. Posey is going to give us a talk on habitat and water quality considerations for southern flounder. Thanks.
Thank you. Well, I appreciate the opportunity to talk about one of my favorite subjects, is habitat. Our lab has, for 3 decades now, been looking at what makes a habitat. What are some of the features that distinguish a preferred versus non-preferred habitat? What's the scale of habitat? And particularly for a variety of nekton, what determines use? Well, first of all, I want to acknowledge that The work I'm presenting today is actually work conducted by a variety of people. Listed there are a number of people for whom they've conducted various projects, independent master's thesis, PhD work, etc. And I'll be presenting bits and pieces of their work. Also, funding. What I'm presenting today is actually coming from a variety of different studies over the past couple of decades. So I want to acknowledge a variety of funding sources, including the Cruffle program. First of all, just a quick reminder of the life cycle. They, of course, have the offshore spawning. They're coming into the estuary. They go up into the lower salinity areas. And then from that, as they mature, they move back down. Some may, as Dr. Sharp mentioned, some may move into freshwater. Some may, according to the literature, stay within the low salinity estuarine areas. There may be some transition. Try this one more time. Okay. So today I'm going to be mainly talking about the juveniles and their habitat. Juveniles are important for most fisheries. They're often a bottleneck. Some great examples within North Carolina, of course, are blue crabs, striped bass, other species, and understanding that juvenile habitat is a key aspect in trying to understand fisheries dynamics and populations. Oops. Some aspects in terms of juveniles, understanding juveniles are particularly important. They're an area where you have high mortality that can affect later fisheries number aspects. Growth during juvenile stages is important in terms of their survival and movement into the adult stages, into the later stages. They're susceptible to predation. Sometimes they're more sensitive to environmental changes and extremes. They're more sensitive to food availability, particularly food limitation. Key aspects of habitat and water quality may have significant impacts on juvenile abundances. And so this stage is one that habitat may be particularly important in later effects on the fishery. But specifics of habitat are often difficult to determine. We don't have a lot of good data. For example, our sampling is often not at scale to really determine fine details and determine fine differences in habitat utilization. Often sampling methods are not really logistically feasible for measuring structural habitats like woody debris, other things you simply can't draw seine through very easily. They may change with growth, and scale can be very important. So, some of our current understanding in terms of juvenile flounder habitat. First of all, as Dr. Sharp mentioned, salinity is a key factor. The juveniles are coming in, early juveniles are recruiting into the low salinity areas. And I just want to point that much of what I'm saying in this slide was summarized very well in the habitat or the amendment for the Fisheries Amendment that was done in 2022, and that graph comes from the Fisheries Management Plan Amendment, summarized there. So salinity, of course, is a key factor moving up into the estuary. Temperature DO, we also talked about, was important in terms of spawning, in terms of timing, and also in terms of juvenile habitat use. Several studies indicate the potential importance of shallow water as a particular habitat, that they're primarily using the shallow edges, but other studies have disagreed with that and suggested deeper water or basically one of the edge areas of deep to shallow water transition. Use of submerged vegetation where present is another aspect we've seen in several studies. This can include things such as marsh edge, submerged freshwater to oligohaline submerged vegetation such as wild celery, Eurasian milfoil, hydrilla, all may be potential habitats throughout the range. Seagrasses, particularly rupia, which is occurring, woodland grass, which is occurring in more oligohaline areas, may often be utilized. However, with that, there's also a debate. Some studies have suggested that they're using the dense beds. Some suggest that they're using the edges. And really, it's the edge aspect, like with a marsh, that's most important. Marsh creeks of various types, Ligohaline, two more marine, are suggested to be important. Once again, primarily the edge areas. Substrate type, some studies suggest a sandy substrate is preferred. Some suggest, well, I get really loud when I get up close to that, don't I? Some suggest that it's more of a silty substrate, and that's probably variation by region. Food availability is another aspect that's considered important in terms of juvenile habitat use. For example, feeding on mysids, some polychaetes, very, very small fish early on, changing to a primarily fish diet later after that. And of course, recruitment can vary with a variety of hydrodynamic conditions that can determine which estuaries where they end up with, and may vary strongly with scale. But what I want to point out with is, showing these little pictures up here, Oligohaline habitats are not uniform. When you talk about they're using these Oligohaline, low salinity to freshwater habitat, you can be talking about sandy areas, you can be talking about cypress areas, you can be talking about woody debris. As you move down to the estuary, once again, your shorelines, your type of habitats available are highly variable. So when we talk about juvenile habitat use in these Oligohaline, low salinity areas, the question comes, what particular habitats are important? Are they using all habitats the same, or really are certain types more important and critical for these early juvenile stages? Okay, some important considerations given this high variability that we may have in habitat within these low salinity areas. First of all, among watershed differences. Do we see differences in habitat utilization in watersheds? For example, in a given system where you have various independent watersheds, do they vary? Within a watershed, do we see variability in different areas depending upon the kind of habitat? Specific characteristics of optimal habitat. All wetland shorelines are not the same. What types of habitats are preferred? Structural habitats versus non-structural habitats. To what extent is that important? How does it change with time? And what is the scale of habitat? We think of Oligohaline as the scale. To what extent are the fish actually differentiating habitats on a much smaller scale? And then, of course, the background then of water quality impacts, which may further change and modify the habitat use. So for the rest of today's talk, I'm going to be looking at these aspects using the Cape Fear River system as an example. Many of the aspects I'm talking about from the Cape Fear should be applicable to a wide variety of estuarine systems. So we're going to look first at the watershed aspect, the Cape Fear versus the Northeast Cape Fear. We're going to look within the Cape Fear system to look at how they vary on that spatial scale. We're then going to be looking at a smaller scale on the order of 300-400 meter scale, protected and exposed shorelines. And finally, we'll be looking at variations on the scale of tens of meters with vegetation and shoreline structural differences. Okay, so first of all, the Cape Fear River. Just a quick orientation. We have Wilmington right out there. We have the estuarine area, and then we have a major split within the Cape Fear River system. We have the Northeast Cape Fear River that comes this direction, and then we have the main stem Cape Fear River that goes up. These are both very extensive watersheds. Obviously, the main stem's more extensive, but they're significant systems. This was as part of a multi-year study. We were looking at nekton in both of these systems over an extended period of time. This was actually a study related to a deepening and widening of the river system. The sampling method we used was drop traps, which allowed us to sample structurally complex habitats. We could actually sample over woody debris and areas like that, so we could actually encompass that component of it. And so what I want to show here is these two stations are on the main stem Cape Fear. These three are on the Northeast Cape Fear. This one is just below the confluence. And then this one down here is located right about there. So a couple facts. First of all, your lowest station, which was a moderate salinity, you're talking about 12 to 15 parts per thousand most of the time. We had almost no catches during the time period we're looking at. But now look at the different watersheds. Within the main stem, these areas, and then after you go for the confluence, for those who are familiar with the Cape Fear, this is Eagle Island, right, just at the confluence. We see relatively high catches. It varies between years. There's significant interannual variability in the catches. That may be interannual variability in the abundances, or it may be simply that we're sampling monthly and we missed their passage through, because this was only monthly sampling. But we see relatively high abundances. Consistently, however, the Northeast Cape Fear watershed has low abundances. We're seeing a significant difference. In this particular case, we have some evidence in terms of why. One of the things I spend much of my life doing is studying worms and the little things that are in the mud. They're really exciting. But anyway, this shows the diversity of organisms, including a number of prey species like mysids, several of the polychaetes and amphipods that are known to be prey within the main stem Cape Fear River system. This is actually one of the same stations as I showed you for the main stem. This shows you the Northeast Cape Fear, except for these two little peaks, which are actually probably not food or deep burrowing polychaetes. You have almost an order of magnitude lower food resources in that tributary. So what's causing that? I could spend probably an hour talking about that. But the key idea is we see these tributary differences, possibly reflecting food resources and other differences that can influence juvenile survival. Okay, now we're going to move from between systems to now looking at primarily just the, it doesn't like me. Okay, there we go. So looking at just mainly the Cape Fear River system. Stations here go from that Eagle Island area where we had high abundances and moving down. Story here is pretty straightforward. You do see a very strong segregation in location. The blue is the uppermost stations, and then the dark blue represents your lower stations. So we see that within this system we get that sort of typical salinity gradient that we've talked about before. We also see strong interannual differences. Once again, it could be that we're missing the passage through of the juveniles because these are only monthly sampling, but they do at least suggest the potential for strong interannual use of these habitats. This I just threw in there. Of course, Southern Flounder is not the only one we get. Summer Flounder picks up here. They're mainly in the lower area with some significant variation. Okay, so now we've gone from between tributary, looking a little bit within that estuarine system. Now we're going to move to a slightly smaller scale. Here what we're looking at is within the estuarine area, particularly that Eagle Creek area, or Eagle Island area where we tend to get high abundances most years. We're now looking at, on a very small scale, of only about 500 to 600 meters, so relatively small scale. We're looking at differences in abundances as you move from the riverine area to just inside protected areas. These would actually be moving sort of outside primary nursery right to just inside primary nursery designated areas. We have a series of these paired samplings. So we're looking at whether or not we get differential use on this small scale as you move from simply the open shoreline, as you may get on oligohaline on down to a more protected area. And what we find is that we actually find, if you look at a variety of nekton, that surprisingly some will stay on the riverine, more exposed area. Other fish will be more in the protected area. In the case of flounder, they're far more abundant within that protected area, just a few hundred meters away from the more exposed area. Not surprising, in some respects, although, like I said, we do get higher abundances of pinnate shrimp and other species within the slightly more exposed main riverine area. So this is a relatively small distance, and so we're seeing a choice over a relatively small distance. I should note this study was conducted in 2021. That was that year of high abundance when I showed you the previous figure. Okay, now we're going to move to the really small scale. So in this case, we're now looking at these various creek areas. Once again, Eagle Island, area of higher abundances, on down. And we're now looking at the choice between vegetation types. We're looking at a scale now of tens of meters. If you look at these systems, and this is very true throughout the state, throughout the southeast, in the illegal haline areas, you get this distinct mixing of vegetation types. For example, in the lower Cape Fear River, or I should say within the illegal haline portions of there, we'll get mixes of black needle rush, juncus, spartina, as well as Phragmites. And as you can see, they're actually often very close together. There's a boat in here somewhere for scale. Likewise, in this one, there's a boat in here somewhere for scale. But you can have them fairly close together. This shows you how close these habitats can be. On this side of the creek, I have Phragmites. On that side of the creek, I have Spartina. Yes, I know Spartina is no longer called Spartina. I will always call it Spartina. You just can't get rid of 3 decades of indoctrination. So you can see you can have very close proximity of these habitats. So the question is, do these various nekton use these habitats fairly interchangeably? Or is there discrimination of habitat on a very small scale that would need to be taken into account as you begin to consider critical habitat type? Okay, well this shows an overall pattern. And I should note that the previous study is being done with fyke nets. Once again, the attempt is to try to miniature fyke nets. The attempt is to try to preserve the natural structure, because all these habitats have a lot of woody debris that's refuge, and we want to try to keep that structure. They're also just not very sampleable by things like seines, because your topography generally goes, oh, knee height, knee height, knee height for me, I'm swimming. So, in terms of the usage, this shows the usage of the various types. You have Spartina, you have Phragmites there, and then you have Juncus. This just shows a variety of species. I'll show the data for the flounder in just a minute. What you can see clearly is that we have a lot of variation in terms of selection. There are some species that very strongly select for Spartina habitat. And I should note this is done over 3 years. This is not a single year study. Select strongly for Spartina habitat. There are a few that prefer Phragmites. Very few prefer Juncus in this particular study. This is all, incidentally, the front edge of the marsh. So we're looking at the fish coming in the channel that are utilizing the areas with different kinds of vegetation on the front edge. Now looking more specifically at Southern Flounder, we find here that over this scale of a few tens of meters, we find distinct differences in abundances. This is a protract measurement where they prefer Spartina, and Juncus just a few meters away, they simply do not utilize those shorelines. They're very much selecting a particular kind of habitat shoreline in terms of where they're occurring most commonly. We do have some data from benthic abundances in terms of food resources, and you do get differential invertebrates occurring within the sort of front areas of these vegetation types, not surprisingly. This is actually just, I threw this in. We've also been looking at some seagrass areas. This is up in Topsail Sound, utilization of mixed seagrass. This is primarily some eelgrass, some shoalgrass mixed in. This is fairly salty and just shows little use by southern flounder there, mainly by other species. Not surprising given the juveniles are moving into oligohaling. Okay, so want to sort of end with talking a little bit about some of the other interactive effects that may also influence this habitat utilization and scaling. First of all, once again, I refer you to the amendment, the Southern Flounder Amendment number 3, the one done in 2022. They have a very nice discussion and this really nice sort of figure that talks about how factors interact and may change habitat utilization. But just some aspects, suspended solids, turbidity can be a significant factor in terms of how you may get small scale or larger scale habitat utilization. Studies have shown that when it's highly turbid, there's difficulty in feeding. They are mostly visual predator. You also get a factory disturbance when you get too much turbidity, and they will actually change their feeding habitat to feeding on perhaps a less profitable prey. You have indirect effects in terms of substrate impacts, changing from coarser to finer sediments. You may change your vegetation type somewhat, may change the kind of silling you have in terms of the edges of the channels. You have impacts on marsh, submerged aquatic habitat. Eutrophication similarly can have a variety of interactive effects with these habitat choices as well. Obviously, hypoxia, indirect effects in terms of food for various species. This actually may be part of the effect of the Northeast Cape Fear River being blackwater. Introduced species and invasive species. Probably the dominant Phragmites that we have in the Cape Fear River is probably mostly the invasive form, as well as other shoreline modifications can all modify these habitat effects. Well, to summarize it, so in terms of habitat considerations, though we have a general understanding of juvenile southern flounder distribution, we're still learning about the details of the preferred habitat. We can see watershed differences based on prey abundance, probably other conditions. We're learning about small-scale differentiation between vegetation types, interactions with other factors. And so this sort of, from a short term, until we learn more about their habitat preferences, argues for retaining a diversity of habitats, linkages and corridors among habitats, until we learn which may be the most preferred type. Thank you much.
Thank you, Martin, and I'll use this microphone because you don't need it. We are pushing into a break, but we can take a couple questions if anyone in the audience has any questions for Dr. Posey. Yes, sir.
Okay. Are you seeing a link in the Cape Fear specifically between the flounder, the health of the flounder's population and the pollution in there?
I think I'll answer that sideways. We're seeing a link between juvenile flounder use and prey and structural habitat. For example, the vegetation choices are correlated with the kinds of prey that are there. More amphipods, we do get more mysids, even on that small scale. Within the large estuaries, it's interesting because the Cape Fear River clearly has a lot more upstream impacts. There's all those animal operations, et cetera. But the Northeast Cape Fear River has got less of that, but it's a blackwater river. So it has all of that natural decompositional component and lower DO. And so it's a water quality issue, but not necessarily one that's caused by human activity.
Thank you.
All right, thank you, Dr. Posey. So now we're moving into our first break / poster session. So I know a few folks got here early enough and mingled and talked to staff out in the hall, but please use this time to go to the restroom, get a refreshment. Staff, as well as some invited researchers, have posters out in the hall just talking about some of the research that we do and some of the sampling programs that we have. So please, I encourage everyone to go out there and find someone and chat them up. And we'll reconvene at 10:45. Thank you. Our next presenter is Dr. Shelby White. She is currently the Marine Business Specialist at the Virginia Institute of Marine Sciences. Shelby has worked with us in quite a few positions, and for whatever reason, we can't seem to keep her. But she's done quite a bit of work, mostly out of Elizabeth City's office, and she's got a PhD in Fisheries Science from VIMS, where she's worked on more of the social science side of it, I think. But while she was with us at Fisheries, she was the lead DMF staff on our satellite tagging project for Southern Flounder, and today she's going to give you a talk entitled Assessing the Migration and Spawning Habitats of Southern Flounder in the South Atlantic Using Satellite Tags. All right.
All right, can everybody hear me okay? Okay. All right, good morning. Thanks, Steve, for the introduction, and thank you all for being here today. So like Steve said, I'm now employed at VIMS, but I was a satellite tagging biologist at the North Carolina Division of Marine Fisheries. And today I'm going to give you an update on one of those collaborative projects between DMF and UNC Wilmington. And this research began in 2019 and was funded by the Commercial Fishing Resource Fund and the Coastal Recreational Fishing License Grant. Okay. So the previous stock assessments identified the need to understand southern flounder migration patterns and movement. There's evidence, like Dr. Sharp said, that southern flounder make long migrations offshore to spawn. And there's evidence that some of these fish remain offshore based on our dive surveys and recreational catches. But there is some questions about the southward movement with spawning, if that's still occurring. And we need to answer these questions to better understand flounder migration and spawning in the future. And like Kathy said earlier, that Director Rawls, that we need to do this for better management. So we still do believe that southern flounder spawn on the edge of the shelf due to some of the ichthyoplankton work that finds them there. But it's important to know, where does spawning occur, really? Is it offshore? Is it near shore or inner shelves? That would be important for habitat management. Also, what happens after spawning? Are these fish coming back into the estuaries where they left? Are they going to other neighboring states in the south? And what habitats are used before and after this spawn occurs? Okay. So the objectives of the satellite tagging study were to identify potential offshore spawning areas used by southern flounder, identify movements prior to and after winter spawning, and identify migration corridors and offshore habitat preferences of southern flounder during this time. So I want to briefly familiarize everyone with the tagging methods that we use, because they are pretty new to flounder themselves. Typically, satellite tags are placed on some bigger species. Some of the tags that popped up, people called and thought they were placed on sharks or turtles or other things that are kind of more that you think about than flounder. But we use the MR PAT tags up at the top, and those are a satellite tag, and then the mini PATs on the bottom, which are a little larger, rounder tag. These tags are both from Wildlife Computers, which is a company based in Washington State. And how these tags work is when they detach from that orange base there, and they float to the surface, and when they're dry or at the surface for a specified amount of time, they communicate with these satellites called the Argo satellites. And from those satellites, the researchers, or who has access to the tags, can pull the data that they collect. So the MR PAT tag is your more basic version. It collects daily minimum and maximum temperature, as well as shows you where the tag popped off. But the benefits of the mini PAT, they cost a little more and they're a little more expensive to purchase and apply. but they do provide additional depth and temperature information, so they collect a lot more than your typical MR-PAT tag. But you can see these tags are pretty large, and if I had one in hand, you would say, wow, maybe you can't believe that fit on a flounder. So we opted to use larger Southern Flounder for this research, just because there's typically a scientific standard where you don't want the tag to weigh so much that it affects the flounder. So most flounder were 19.5 inches or larger, and this also increased our chances of finding female Southern Flounder, which is really what we wanted to focus on for the migration. And you may say, why female? Well, we don't know much about the males yet, and it's kind of hard to identify. It's impossible to identify without genetics and some additional testing, whether it's male or female in the field without sacrificing the fish, which we obviously do not want. And then all tags, all fish, which I'll show you in the next slide, were tagged with a high reward spaghetti tag. So Amy Staples will give you kind of the rundown of what the spaghetti tags look like, but really it's like attaching a spaghetti noodle to a fish. And so before we put these tags in the natural environment, we conducted experimental tank trials on these tags to make sure we were using the best method to have the highest retention once the fish was out in the open ocean, and the best healing rate. So we didn't want anything really to interfere that we could control with these tags. We wanted the fish to heal. We wanted them to complete their migrations as they had already planned and to go on so that we could get that data. And so from this trial, it was determined that the satellite tag would be, that did not turn, placed in the upper middle portion of the fish and attached using that spaghetti tag noodle. And then also, again, there's the high reward red tag at the end. And so when fishers call in this information, if any of you have ever seen one of the captured or rekept a tag, you call in that information and there's some monetary reward associated with it. So that gives us, once the tags pop off, another piece of information that we can use potentially to see where these fish are going. And the tag that went through the middle of the fish was secured by a blue backing plate. And this was because in the tank trials, you can imagine the flounder laying on the bottom or moving around and the tag would rub and pull through, and we did not want that to happen and the satellite tags detach prematurely in the wild. And again, the high reward red tag is another additional source of information that is useful for us. Okay, so this table, it looks like a lot, but it's not. This experiment lasted for 3 years. So we started in 2019, we did the tank trials, put the tags in the water in 2020 all the way through 2022. Typically in the fall when the migration was occurring. And in the first year, we released 130 tags, satellite tags. All of these were MR-PAT tags, and so I do want to highlight that this table is really focused on the MR-PATs, and towards the end of the talk, I'll give you a little more detail on the mini-PAT tags, which are still being looked at. In the first year, we tagged in 3 different locations, and this is a learning process also for us. So communication time with the tag satellite is when the tag pops up, and it says, this is where I'm located. You can adjust the time on that. And so flounder, we found we had them set at 24 hours and found that flounder like to go in kind of the shallows and hang out at night. And if the tags pop up and then they pop off where they were already tagged, then that's not giving us much information, right? So we also adjusted the release patterns. So in the first year, we used 15-day intervals all the way from January 1st to March 15th. And we thought maybe we could catch the fish as they're coming back in or going out, and we'll know exactly where they are. But in the second year, we improved that methodology because we thought maybe we had missed some of the fish coming back in, and you'll see from the results what I mean by that. But in the second year, we also partnered with South Carolina Department of Natural Resources and Georgia's Department of Natural Resources. Although we weren't as fortunate to find as many fish in those areas, they both received a tagging kit, tagging video, and were able to get a couple tags out. So that's why you'll see the 5 locations in year 2, 2 of those were in South Carolina. We changed the communication time to 8 hours, and this was because we had a couple people say that they found the tags washed ashore in Hatters or on the beach elsewhere, and they looked like they had a little bit of fouling on them. And the longer the tag's fouled, maybe it stays wet and then it doesn't communicate properly. So we wanted to prevent all of those issues the best we could. So we coated all of the tags with anti-fouling paint based off Wildlife Computer's recommendation, and we're able to see an improvement with tag communication. And I just want to highlight, you can imagine putting technology in water and then letting it go to the ocean. And so there's always some things to learn and some things to adjust with that. And so another thing we adjusted was the release pattern. So when the tags were set to pop off on that predetermined date. So we did a 10-80-10 distribution. So really 10% of the tags were released in early December, 80% between mid-December and January, and then another 10 at the beginning of February. And again, this was in hopes of kind of getting a more randomized sample of when these tags were popping off and where the fish truly were. And the sampling gears we used for these have varied throughout the years as well, based on some changes in the fishing season and kind of the fish availability or who was fishing. So we primarily worked with commercial pound netters for this, and then we used some of the independent sampling gears from the South Carolina and Georgia DNR. We used gill net and hook and line and worked with some charter fishermen down there as well to figure out how we can get these tags out. So this is just a map showing you where things were tagged across all 3 years. So just want to highlight, we were in the Albemarle Sound and then did some in West Bay in later years or in the last year of the study. Hatteras, Core Sound, Mason Borough, Ocean Isle, the Lower Cape Fear, and South Carolina. So we were all throughout the state and into other states as well. So once the tags are deployed and they all released on a predetermined date, we are able to access that information and download it from what the tag saw while it was out in the water, potentially. And this is also the degree of error associated with those locations. So when a tag pops off, you can see, looking at this, that there's varying degrees of how many meters away that the tag may be or the fish really was from where it said it was. So we decided to go through the data and make sure that we were using the best possible tags so that we didn't have any biases. But the numerical classes are more accurate than the ABs or Zs, and the ABs and Zs don't provide any accuracy estimation, so you can imagine that that doesn't help much if you want to know where a fish was. And this is consistent with some of the other work that's been used for satellite tags, following the method of finding the first class 3 that occurred within the first 2 hours of the tag popping off. That's your absolute best location that you're going to get. And if there was no 3 within that first 2 hours, we did the next numerical classification within the first 24 hours. So as long as it was numeric and within 24 hours, those were tags that we felt accurately represented a better depiction of where the fish really was. And so after cleaning the data, we had a total of 171 locations out of 257 tagged fish. And you may say, well, that's a few that didn't work. But again, putting tags out into the open ocean and kind of seeing what that environment's like, and anything can happen to these fish. They could be prey items for another larger species. The fish could suffer mortality. So we're working with a lot of different factors, and this is actually pretty consistent with what people see with satellite tags. So these are some of the results that may be interesting to you all. This map is a depiction of the satellite tags based on pop-off locations by where they were. So we considered the inshore environment within the estuary if the fish didn't go outside of an inlet and remained where we had tagged it or somewhere in that system. The inner shelf is delineated by if it was 50 kilometers or less from land, and then the outer shelf was indicated by more than 50 kilometers. So inner shelf is in green, or inshore is in green, inner shelf is in orange, and outer shelf is in blue. And so there's a lot going on here, right? We thought all these fish would be offshore, we would know where they were, what habitats they were using, and the problem would be solved. But you can see some of them remained inshore. And is that because they weren't migrating this year? Were they waiting for something different to leave? Did we possibly disrupt their migration patterns with this tagging? The inner shelf, that is also another good question for the orange ones. That's not quite all the way offshore, but that is where the tags are popping up, and they're all at different times. So it kind of made us think, well, are they staging here and really just waiting for the better environmental cue to say, hey, it's time to go offshore, the rest of the fish are going, or something along those lines. But what we're not seeing are those long southward migrations. And that's not to say that they don't happen, but it doesn't appear to be happening during the suspected spawning period that we were able to observe. And so I do want to provide one exciting piece just to highlight the importance of potentially turning in spaghetti tags if you find them. So we had an instance where we had tagged a fish in Core Sound, there, and that fish had migrated out of the inlet. The satellite tag popped off near shore, the inner shelf, and then that fish was recaptured about 9 months later inside the estuary again. So we are seeing some evidence that they're coming, or this particular fish came back to the estuary where we originally tagged it. Okay, so now on to the mini pats. These were deployed at the same time as the MR pats, and they function very similarly other than providing additional information. The tagging methodology was the same in the center of the fish, the high reward tag near the tail. But I do want to show you some of the results from the mini pats, and I will do this highlighting a couple that were tagged in Core Sound in year 2. And so this is where this fish I'm going to be talking about was, where the black circle is. And I want to direct you to the gray figure on the left to start with. The date is on the bottom, and then the depth is on the y-axis, so right there and right there. And you can see that the fish, ignoring temperature, because temperature is a little more mixed than we thought it was, that fish stayed at a pretty stable depth for quite a while, from November pretty much till January. And then all of a sudden it dives down, kind of comes back up, dives down again, comes back up, dives pretty far down to about 30 meters or 90-ish feet. And this occurred a few times. And you can see it even better on the right, the figure on the right that's in the blue color, those points where they're going up and down in the water column. And we think these are indicative of spawning rises, so the fish is actually releasing its eggs and spawning. And we see this in other flatfish species. So this is really the first instance that's been highlighted in Southern Flounder. And again, I just want to give you another example. We did see this a couple times, so it's a little more broken here, but if you go to the blue, you can clearly see those peaks, those rises and those falls. So of the 34 mini pats that were released, 7 exhibited these spawning rises, and that's pretty cool to see. These rises ranged from times of 5 minutes to 195 minutes, so almost 2 hours worth, that they were going up and going down, but an average of 37 minutes. And they span depths of 5.5 meters all the way to 21 meters, but an average of about 13 when they were going up and down. And on average, fish had 6 individual rise episodes, so that up and down peak. So I just want to put this all together and kind of summarize what we learned, what's next, and where do we go from here. What we learned is that it looks like a lot of fish are maybe using that mid-shelf habitat, about 15 to 30 meter depths, which is a little different. Like Dr. Sharp said earlier, we really thought it was more offshore, and that's not to say it isn't. It just isn't what we necessarily saw, and that could be based on the timing of release of the tags. We know their offshore migration is fairly short, so maybe we missed that point and they were already on their way back in, or the spawning rises kind of indicate differently. The timing of migration to offshore areas varied, so we typically tagged in the fall, like I said, all the way from September initially to November. And so they were leaving the estuaries all the way through November to January. So some were late, some were early. It's a little bit variable, and maybe that's a pickup from the environmental cues or kind of the latitudinal change from north to south. Like I said, 7 individual fish had spawning rises occur, but with variable behavior. So it seems like in general they're doing the same thing, but this could take a short time, a long time. The depth may not necessarily be the same. And there's no evidence of long-distance southward migrations based on what we were seeing. Again, that doesn't mean that some other aspect of that was not captured and that it's not occurring, but it wasn't for this. And so what's next? We need to still investigate the migration behavior. So one of those things that I talked about was potentially understanding the impact of tagging on the fish. You're putting a couple holes in a fish. Now, while they healed well in the tanks, and we believe they healed well outside of the tanks, that doesn't necessarily mean that they didn't say, whoa, I'm not going offshore, like something weird's happened to me, right? So thinking about what that effect is and how that could impact and maybe how we can improve that system. We could also consider possible offshore histology studies. Now that we have a decent idea, at least from this evidence, of where some of the fish are, could we understand spawning masses and ratios and what we're really looking at and how to get those behaviors together? And also, this study focused on females, but we could very well investigate male behaviors and migrations. It would take a little bit different approach. But understanding what they do, maybe they're the other half, and they are the other half of the puzzle, but one that we're not so clued in on. And so with that, you're fine. I'll just say thank you to the Commercial Fishing Resource Fund who funded the division's half of this research, the Coastal Recreational Fishing License Fund, which funded the UNC Wilmington portion of this research. Thank you to all the commercial and recreational fishermen that collaborated and helped with this research. It really would not have been half as easy without their help. And thank you to the DMF and UNCW staff who very willingly woke up very early and tagged lots of fish and had some long days out on the water. And at this time, I'm happy to answer any questions if I have time. Good?
All right, thank you, Shelby. Does anyone have any questions? Rebecca.
So, great talk, and lead into some of the stuff I'll be talking about. Great talk. It's a good lead into some of the stuff I'll be talking about next. We're actually going to talk a little bit about offshore histology. So, nice intro. I was curious about what time of day you're seeing the spawning right?
Oh, that is a good question that I do not know the answer to, because I would have to go back and look at that right off the top of my head. And Dr. Scharf may know that. He was a little more involved with the mini pats than I was. But that is good. I'm not sure if it was morning or night, but that may be an interesting.
Just curious.
Yeah, that's a good question.
Also, along those same lines, of the 7 fish that you saw the rise occurring, what was the max depth? Did they exceed 30 meters?
I don't think so. I think that was because the average was around, I said, but the ones that we looked at, maybe they went slightly beyond that, but not enough to say that it was substantially different. Yeah, so about 30 minutes. That's why we suspected that the mid-shelf habitats were more utilized, which is an interesting finding compared to the offshore.
Very. So, and one more question.
Yeah.
The fish that you had the tag release, got the data, satellite data, that was caught inshore again. Was that the tag return from the $100 tag?
Yes, it was. Yes, so that was a fisher who called in the $100 red reward tag. Yep. So that was the third piece or the second piece that it worked out a couple times, and I want to say other people have found the satellite tags washed up, but they don't have any monetary value, so usually those are just, and it's hard to say when they washed up and if they went to where they were supposed to, but yeah, the high reward tags and even the low reward tags are very effective in giving us some of that info.
Great call.
Yeah, thank you.
Okay, hi. Great project, I love it. I was just curious what you think the most important data output that you got from this project as compared to conventional tagging methodologies.
Yeah, so one of the most that I would think of immediately is that we don't typically get tag returns offshore. That seems to be more of a kind of inshore estuarine Southern Flounder fishery. That's not to say that there aren't people out there, but it's just not as common. So that was a great portion of that, and something that we couldn't have gathered. And also, these tags, the high reward tags, stay out for a long time. So we really kind of got a short picture of what that fish was doing, whereas with the spaghetti tag, maybe that fish was at large or out in the water for years before someone caught it, and then we kind of missed some of that story within it. So there's definitely benefits, and the mini pats are even greater. Like I said, they cost quite a bit more, and they're a little more scary to lose in the ocean, but they do provide, the timing you set it to. We collected data every 5 minutes on temperature and depth. So that's great to know where fish are in a place that's not typically as easy to sample.
Thanks. So I'm trying to reconcile your talk with what Fred showed first. So what I saw from Fred's talk was the immigration of fish happens really quickly and very consistently. And it seems like the data you showed don't really directly match up with that. And I'm trying to do some interpretation, so tell me if I've got some of this wrong. And then also Fred showed pretty good mixing to the south, which your data didn't necessarily show. So how, as a scientist, are you reconciling those differences?
Yeah, that's a great question. So Dr. Sharp's data, was really the timing and where they go kind of informed how we set up this study. But like I said, since the tags only provide, the MRPAT tags, which was the majority of our tags, provide a singular location, we don't really get some of that whole story or the year later recaptures from that conventional tags. It's possible that spawning is still happening fast. So maybe once they're inshore, aside from the ones we saw the spawning rise on, maybe they've already made their offshore migration and have already come back in or to the estuary. The migrations were all the way from November to January. So if it's only a few days and we had the tags scheduled to pop off in January, but it was a November fish that had left, then we very well, missed that part of it. And then the southward migrations, that is a puzzle for us as well, because we really thought we would see that, and we thought even more so with South Carolina and Georgia being involved, that maybe, then we'd really have an understanding of where fish were going north or south. So that is an interesting point, and maybe it's something that happens at a different time. Maybe it's a different males versus females or something going on that we just couldn't see with the satellite tags. But those are all very good points, and they have absolutely puzzled us for a while as well.
That's the main one.
Hi, thank you. I wanted to ask a question about, not to compare apples and oranges, but how does this data reflect with the summer flounder? What studies have been done to see, wow, one species is declining and another species is at least stable. What's the difference between the two species and how much information has been captured to compare the two species?
Yeah, okay. Well, I'll answer that the best that I can with what I know. So Southern Flounder are kind of a more recent fishery and kind of a growing recreational fishery within the last 10, 20 years or so. So I think we're up and coming on understanding that, and North Carolina is more or less working to lead that research in that area. So how it compares, I don't know if I could totally answer that, because they are they're similar, but they're very different species and occur in different ranges. And, summer flounder, we're seeing kind of that northward movement just because of environmental changes and where they're going. But with flounder, we don't necessarily have that information quite yet, I don't believe. So we're working towards that. We're at the kind of cusp of understanding flounder better, if that helps. Ish. Thank you.
Well, we can take one more real quick.
Yes.
Hi.
Hey, great talk, Shelby. This is Joe Luscombe at GCU. I wonder if you're planning any future studies, or your group, to go beyond March for release times, because I believe the southern movement happens after March. And you'll hear in our next talk, fish are detected to the south in August. We don't really know where they are with the acoustic tags that we use or your satellite tags between March and August. So are you thinking about repeating this with longer release times?
So I will highlight that the mini pad tags released by UNCW were extended beyond our typical March cutoff. So I want to say that some of those in the first year, the second year that we used them, it was to mid-April. And then in the third year that they released them, it was all the way till July. So that is capturing the spawning rises and where they popped off. And so I would defer to Dr. Sharp, to see if there was any interest later on in potentially the timing or where those tags were released. And as far as the Division of Marine Fisheries, I will defer to them. I don't have a say-so so much in that anymore. Thank you.
Thanks, Ray.
Thanks.
All right, great talk, Shelby. All right, next we have Dr. Rebecca Ash. She's the Associate Professor of Fisheries Biology at East Carolina University and the University Director for the Interdisciplinary Applied Data Analytics Certificate Program. And I did have to Google that last night to figure out what that was. But Dr. Ash holds a BA in anthropology from Smith College, a master's in biological oceanography from the University of Rhode Island, as well as a PhD in biological oceanography from Scripps Institute of Oceanography at UCal San Diego. At ECU, her research focuses on how climate change affects fish populations with a focus on questions that advance our ability to manage fisheries in a changing environment. And today, her talk is entitled A Multi-Pronged Approach to Investigate Offshore Migration and Habitat Use by Southern Flounder. So, thanks.
Okay, welcome everyone. I'm glad to be here. As mentioned, I'm Rebecca Ash. I'm coming from East Carolina University, and we're going to be talking about offshore habitat use by southern flounder. This is really going to complement the talks by Shelby and Fred quite well. And because of that, they have gone through a lot of the background information, but this is just a review. A large number of fish in this ecosystem use estuarine habitats as juveniles, and then they migrate through inlets to go offshore to reproduce. Because we don't have fisheries offshore, oftentimes there's huge unknowns in terms of where they're going and what they're doing when they're offshore. So this is true of southern flounder as well as many other fish. We have also been hearing about for the last couple of years how this is a very valuable fishery. So this is data from NOAA from 2019, but unfortunately it's been classified as being overfished and experiencing overfishing. So this is information from kind of some of the stock assessment reports that have been put out. And as a result of that, there's been basically kind of declines in the amount of fishing allowed because of regulations. But like one of the questions, as I mentioned, is we don't really know where they go offshore, which has a question, leads to like whether we should be managing them at state level or more cooperatively across many states. And this is one of the studies that Fred presented earlier by Verena Wang. This is data showing you basically different genetic populations of southern flounder. The red are one population, the blue are another population. And what you can see is throughout the South Atlantic states, most of the data are blue, indicating that there's one population and that there's a good deal of intermixing between states. Suggesting that like we do need to coordinate with management potentially. However, as I mentioned, we don't really know where they spawn. Fred did an excellent job of reviewing the historical data from this. This is all from the 60s up to the 90s. There has not been any offshore work studying this species largely for decades. From that previous work, early stage larvae have been found on the outer continental shelf. This is have some maps showing those locations. So we thought at the beginning of this work that they're going to that outer continental shelf habitat to spawn. We decided to look at this using a multi-pronged approach. I'm going to be talking about all 5 of these methods, so this might go a little quickly. We used a different tagging method, acoustic tagging, very similar to some of the stuff Fred mentioned, but looking at more offshore habitat. We modeled where larval fish came from to backtrack where their spawning sites might have been. We did some offshore surveys of fish eggs and then barcoded those eggs genetically to figure out what species they were, because it's really difficult to identify eggs based on just their morphological characteristics. We looked at the chemistry of fish ear bones, which can provide some clues about what habitats they were inhabiting in the past. And then we did some basic biological research on age, growth, and maturity just to contextualize all this. So that is a lot of different methods. I could not have done this work alone, so I do want to highlight that this was a team project. Many of these team members are in the audience. I'm going to be specifically highlighting a lot of the work done by graduate students at East Carolina University. Beyond the people shown in this picture, we had funding from a Cruffle grant. I used some Sloan Foundation early career money to supplement that Cruffle grant. Also, we had help from people like Mike Loeffler, Fred Scharf, Jeff Buckle, all providing scientific advice that helped guide the study. There were fishermen from Pound Netters who helped us, so that included the Craddock family, the Willis family, and Zach Roman. There are about 7 undergraduate students who contributed to this research beyond the grad students. A lot of members of my lab who are studying different things but came out and helped us with field work in ways that were very helpful. So I just want to acknowledge all those people who made important contributions. Okay, so I'm going to start off with talking about some of the work of Justin Mitchell, who is a master's student. And at the same sites where we tag fish, the smaller ones were taken back to the lab to look at age, size, and sexual maturity. So one of the things that Justin did is he took basically ovarian tissue from female southern flounder and looked at it under a microscope under these slides to figure out what their eggs looked like. And what you can see from that slide is the eggs don't look uniform. They're different sizes, different shapes, which indicates that they're at different stages of development. What this means is they don't spawn just once. They do what's called batch spawning, where they might spawn multiple times over a season. So that's like a first piece of information we were learning from this study. Justin also looked at basically the size of the gonads relative to the size of these fish. And from previous work in the Gulf of Mexico, we know when basically that exceeds 1% of their body mass, that's when these fish are starting to spawn. We sampled this throughout the fall, a little bit over the summer, and we found consistently across both years of our study that late October to November is when their gonadal biomass exceeded that limit, indicating that they were getting ready to prepare for spawning, and this was usually right before the immigration timing. Another thing that Justin found for this is he classified fish based on kind of these slides into different stages, and that we found that basically the 50% mature age was at age 1, which is similar to what other people have found in the past. We also looked at size-related patterns. So as Shelby mentioned, the larger fish tend to be female fish, and based on the size, we know that all the fish that we tagged, which were the larger fish, to guarantee that they were going to go offshore towards their spawning habitat, did tend to be female fish based on kind of Justin's work. We also use that to look at basically relationships between timing of 50% maturity and size, and this is Justin's results compared to previous studies. That first study in blue was done with a different methodology that's less precise. It indicated that fish were maturing at a smaller size, whereas Fred's work by Midway and Scharf indicated that they're spawning a much larger size. The next study by folks at DMF indicated that they were spawning a little bit smaller, and Justin's work is indicating that they're getting to like this 50% size of maturity at a smaller size. So this looks like this might be a trend, with more recent studies indicating that they're reaching maturity at smaller sizes. There was never any previous work on this on male fish, and we had a relatively small sample size of males, but based on this, it looks like 304 millimeters is the size of 50% maturity among male fish. Okay, so I'll get into our acoustic tagging methods. We had a sample size of 210 fish. They were tagged in Albemarle Sound, Pamlico Sound, and Core Sound. And we had an array of receivers at different locations to track where those fish went. At 21 different locations, we had inshore receivers. These were placed at different inlets the flounder might migrate out through. And then we had an array of 20 offshore receivers that were spaced 1 kilometer apart. Initially, that was at Cape Lookout, and then in our later part of the study, we moved that down south to Frying Pan Shoals. Those acoustic receivers are static, so you only find out about like where a fish is going if they pass by them fairly closely. So in addition to that, we used a wave glider, which is this roving underwater robot to look at spatial patterns in a little bit more depth. So what did we find from tagging? So I do want to highlight we have a couple posters, so this is just kind of a quick overview. But the first thing is we thought at the beginning of the study that there was one answer to where the flounder went, and we've learned that it's more complex. So when they're exiting basically the estuaries through inlets, even if they're tagged at the same location on the same day, they will exit through different locations as shown on this map. And this is work done by a research technician, Caitlin McGarrigle. Another thing that we found is that they're leaving a little bit later than what Fred had found in the New River. So we were finding basically them leaving inlets between mid-November and also mid-December. We looked at this 2 years, and it was pretty consistent for our estuaries across those years. So this might be a spatial difference since we're tagging them a little further to the north. So another thing that we looked at is basically are fish going offshore to this hypothesized spawning area. And we did detect a couple of fish in that hypothesized offshore spawning area. So this is one example of that. But many other fish were doing what Shelby saw, using coastal oceanic habitats that were typically 15 to 20 kilometers from shore. So this is our offshore array, and the red areas are showing you where that fish was hanging out, so closer to shore than we previously thought. We also found that a number of fish went offshore during winter, and then we saw them again at inlets, typically the next spring, often around March. So there's a couple examples of this. So this is another example of a fish that was detected offshore and then was then seen again at an inlet the following spring. Here's another example. And then this one is one that I actually think is a really cool example. This is over 2 different years, the same fish. So it went offshore, was detected at inlet, and then the next year, we didn't see it exit or go past the inlets, but it was found offshore again on our array. So this is kind of suggesting kind of seasonal patterns across years. We found that some fish likely overwinter near inlets, so there are a handful of fish, not that many, but that we would see consistently throughout the winter at some inlets, particularly Barden's Inlet, which seems to be a particularly important location based on where the fish were exiting. So Joe Leskovich and Tyler Peacock have a poster, but they've been looking at more synthetic patterns across the different species. And from this graph from them, we did see that some fish, rather than going back to the inlets, migrated further south. So we saw fish going southward towards the southern part of Onslow Bay, including the Cape Fear River. We saw fish going towards South Carolina in the Cooper River and the Savannah River in Georgia. So this is a little bit more of that synthesis from Joe and Tyler, and this is showing you all our tag returns that we've seen, or tag detections from 2020 to 2023. And what I want to highlight here is most of these were from North Carolina waters, although a couple of them have been found further south. When you look at directions of movements, most of that direction is towards the southwest. However, this is a plot of each individual fish that we saw movement data, and it's showing you lats and longs of where they are found, so it's really hard to see. There's a bigger version of it on the poster, but what you'll see here is a lot of them look like just dots, so those are fish that did not move a lot, where the ones that look like lines are fish that tend to have more movement. So what we are finding so far is that 75% of the fish or so basically are retentive. They're not moving all that much, where about 25 of them are undergoing longer range of movements. So I'm going to move on to another component of our study. This is larval dispersal modeling done by Brian Bartlett. So there is a long-term time series in Beaufort run by NOAA that we've contributed to, where they basically during fall, winter, and spring will collect larval fish. We collaborated with them from several years, looked at larval flounder, and then aged those flounder based on their otoliths or ear bones that have daily growth rates or daily growth increments. From that, we knew how long these larvae had been in the water before they entered the inlets, and then we could take data from a couple of different models that utilize information on currents to backtrack, given those currents, where those larvae would have come from at the time when they would have been born. So that's the work that Brian did. He looked at a couple different years, 2005 to 2006 and 2018 to 2020. Those were selected to be both more recent and a little bit in the past so that we could look at basically did patterns change over time as this fishery has declined. Okay, so this is Brian's results. These are maps showing you probability of larval origin. So this is based on that model where the larvae would have come from based on currents. And anything that's dark blue, the larvae could have come there, there's a low probability. Anything that's kind of green or yellow or orange, that's a higher probability of a site of origin. And what you can see is across every single year, there's this hot spot in southern Onslow Bay, kind of near Wilmington, where we get a higher probability of larval origin. So these are annual averages. And if you look at kind of basically daily pathways, sometimes you see some more variability. So these were rare, but occasionally you would see these episodic connectivity where the larvae were coming from much further distances from places like Georgia, South Carolina, or Florida. So this is kind of reconciling some of the different results that we've been getting and is really consistent with a lot of the patterns we're seeing from the telemetry. In addition to this, we did offshore surveys looking for fish eggs. These were done in 2 years, February and March. In 2021 and 2022, we collected about 100 samples of ichthyoplankton or larval fish and fish eggs. And from that, we found over 10,000 fish eggs. We then basically sequenced about 1,000 of them using the CO1 gene, which is often referred to as the barcode of life. It's a gene that's basically used to identify species. The maps are showing you our year one data sampling locations. And during the year one, we did find flounder at some of our most offshore sites. There was only one egg identified, but this does confirm that they are actually spawning in our offshore waters. Year two, because of weather, we couldn't travel quite as far offshore, but we still did surveys. We actually did more surveys. And surprisingly, we also found eggs, but they were much closer to the coast, a lot of the areas that Shelby mentioned. So this is suggesting that there is probably spawning closer to shore as well as in these offshore areas. We found 4 eggs in this year, so we do have confirmation of spawning in North Carolina waters from this. Paul also found a variety of different species. So this is our year one results, just showing you the diversity of species across stations. I want to tell you a couple highlights because these were cool and unexpected. The 2 most abundant groups were spot-tailed pinfish and sea robins. Neither of these are commercial fisheries, but they were really abundant in terms of what's spawning offshore. So this is just an aspect of fish biology that we don't know about and maybe needs to be investigated, because these might be important forage for some of the species that are commercially important or recreationally important. Okay, so lastly, I'm going to talk to you about the chemistry of these fish ear bones and what that can reveal to us about offshore migration patterns. So otoliths form these daily growth rings, and they're mostly made of calcium carbonate, but they'll incorporate other chemicals into their otoliths. What chemicals they have in these trace levels depend on where they reside, because they're reflecting the seawater and its chemistry of where the flounder live. So we looked at about 300 fish, and a variety of chemicals are listed here. These samples were processed at a geochemistry lab at University of Texas Austin using some really complex equipment. This whole room is the setup of all the machines that they were using. And essentially what they do is they shoot lasers at these fish ear bones. The lasers will ablate or destroy the ear bones, and then that destroyed material will be kind of analyzed for basically chemistry based on these instruments. So my alternative title to this talk is Shooting Lasers at Fish to Detect Their Darkest Secrets. Science is Magic, because this sounds crazy. If you Google this title, this is what Google will tell you that I'm doing. I'm not quite sure why there's lasers coming out of that duck's eyes, but I found that kind of funny, so I wanted to share that with you. So this is our results. So the first thing that we found is that there was one chemical in particular that seems like it's most important for figuring out where these fish were in the past, and this is strontian. So there's a strong relationship between strontian and the salinity in the water. These are averages from each of our sampling dates, but you can see that basically the more saline environment is, the more strontian that gets incorporated into the otolith. There is a good deal of variability, though, between each individual fish at these sampling locations. And from this, we can look at how much strontium there is at each segment in the otolith and kind of have a sense of, was this fish in the past in water that's salty versus fresh? So this lets us backtrack where fish were. And these are two fish caught from the same day, both from Core Sound. And what this is showing is that they have really different histories in terms of the salinities occupied. The fish on top, Core Sound is the saltiest water that it's ever been in, where the fish on the bottom basically has been in saltier water in the past, suggesting that maybe it went offshore, spawned, and then came back to a more estuarine, fresher environment. So we looked at this across about 300 fish. These are our results for age 2 fish, comparing females and males. And anything beyond that, like a red line, indicates that it likely was in offshore waters based on kind of average conditions. Although, again, there's a lot of variability between fish. So based on this, this is suggesting that between 25 to 40% of the fish that we found and collected in the sounds may have been offshore in the past at some point, likely to spawn, but that a lot of them had never been offshore before. So one other thing that we looked at is kind of maturity stages and whether fish had indications of past spawning based on kind of looking at their ovaries. So these are, again, slides from an immature fish compared to a regenerating fish that previously spawned and is kind of regenerating after this experience. Javon Smalls did a lot of this work as an undergrad intern. He's from Washington College. And this is what we found. So we had collections of fish both in fall and summer. And our summer collections were in Pamlico Sound and Cape Fear. And the only place we found large numbers of regenerating fish were our offshore sites sampled offshore of Cape Fear. We had summer samples from the same season in Pamlico Sound, but found very, very few regenerating fish there. So this is suggesting, again, that fish probably are staying offshore in the summer after spawning rather than going back to the estuaries, because otherwise we would have seen more fish in that regenerative stage in Pamlico Sound. I do want to just highlight some of our posters. So Brian has a poster on the larval dispersal modeling. Paul has a poster on the DNA barcoding. He's not here, so if you have questions about that poster, I'm the person to go up and talk to about. Tyler has more details on some of the acoustic telemetry and migration patterns. And I just want to highlight to you my conclusion. So we found that flounder are exiting the estuaries through diverse pathways. We found that some of them are going to this offshore continental shelf habitat during the winter, but more fish are staying in coastal oceanic waters, very consistent with what Shelby presented, so complementary, but using different data sets and tagging methods. From DNA barcoding, we found the same thing. We found some eggs offshore, but more eggs closer to the coast. When these fish go offshore, some of them seem like they go back to inlets. I saw them migrate further south. That seems to happen over summer months. And then there was a small number of fish that seem to overwinter near the inlets. And as far as we can tell, they may not go offshore, but it's also possible that they could sneak out and come back undetected. But they appear to overwinter. And that's also been seen recently in Texas Southern Flounder, that there's contingents that are non-migratory there. For otolith microchemistry and the microscopic examination of otoliths, we found that it looks like more fish stay offshore after spawning because we didn't find a large number of the older fish caught in the estuaries had this indication of being offshore based on their salinity patterns. There is diversity there. And from larval dispersal modeling, we're finding that a lot of the larvae seem like they're coming from further south, but from southern Onslow Bay. Periodically, you can get this greater episodic connectivity between different states, which might be sufficient for maintaining some of the genetic diversity, even if it doesn't happen all the time. So with that, I'm going to see if there is time for questions, and this is some of the fieldwork pictures from our team.
All right, that was great. Sorry I didn't get up for your 5-minute warning. You drew me in so much. It's such great work. But we can take a few questions. Got a few. So. Whoever gets there first.
That's there we go. With the studies with the eggs found offshore, were any other flounder species detected in those studies, like the summer Gulf?
Yes, we actually had more detections of Gulf flounder than Southern flounder. So we actually were finding quite a lot of Gulf flounder.
Okay, so how much money would it take for you to answer all these questions?
Oh my God, that's a hard question. Yeah, so this was a grant that was about $500,000. So that kind of gives you a sense of like what you're able to get for that amount of money. And hopefully we delivered well on that money. I think like for my perspective, like some of the biggest things that I'd like to see happening is I was really excited about the offshore DNA barcoding results, so I'd like to see some more studies looking at that. I actually am working with some people from Wilmington and some people from Chapel Hill to put in grants related to doing that, not specific to Southern Flounder, but we'd likely learn more about that. I think that also, like, I think the offshore histology produces some really interesting results, so I think that like we really should be like maybe focusing on some of those hot spots to better detect basically spawning habitat in some of those areas. The acoustic tagging is cheaper than the satellite tags, but there's pros and cons of each of those techniques. The acoustic tags, you can get more locations to get migration routes, but it relies on fish kind of migrating past receivers, so there's some fish we just never know where they went because they maybe never went close enough to a receiver. So I do feel like they're complementary. It's hard to put a price tag on it. I would have to think more about that, but kind of maybe gives you a sense of scale by just knowing how much we've spent so far.
No, I think that's an incredible return on the amount of money for sure. So the last thing I have for you, what percentage of fish would it take migrating south to explain the lack in diversity, how we basically see it as one stock? And is that significant enough to suggest that we have to manage across all, or that our local efforts are significant enough to guarantee our future?
That's a really good question, and I'm not a population geneticist, but like my understanding is sometimes low levels of exchange in genetics can lead to kind of panmixia, because, you know, that even if there's a change every couple generations, that can mean that genetically the stocks are quite similar. And my hypothesis is that might be what's going on here, but I do feel like it needs more research. I do think like the way that the state's been handling this is probably a good balance, where they're focusing on doing management at the state, but consulting with people from South Carolina, Georgia, and Florida. So it seems like a good kind of balance of the two of them. You know, I think in terms of like what should be done with management, whether state and regional, that's definitely a question that is going to take a lot more stakeholders into account. And I think that maybe you should ask that to some of the people from DMF later today as well.
Okay, thank you.
All right. Oh, one more, and then we'll go to lunch.
Okay, I just have to follow up on that. So, does the totality of the science from your professional perspective support a coastwide management of this species?
So that is a good question, and I've gone back and forth. Like my very early results were initially suggesting that we're seeing most of this fish in North Carolina waters, but as we've gone on with the study, we found more from Georgia, South Carolina, and I think that that's because it might take some time for these fish to migrate down south. The other thing is we're dependent on people detecting our fish and uploading the data. From this work and some work that I'm actually doing on a different project, I think North Carolina actually might have better returns on tag detections and sharing data than other states. Some of the other states seem like they kind of keep their data a little bit more closer to their chest. So I think a question that I don't know the answer to is maybe more fish are migrating, but we're missing them from South Carolina and Georgia. So right now it does seem like that we could like kind of manage things maybe at a state level, but there's also a lot of unknowns. So I think that communication is going to be essential moving forward and continuing to investigate this. So couching my answer, but like it's something like where my thinking of that has actually evolved recently based on like our new results that we've got in the last couple months. Okay, I'm off the hook. Oh, one more.
One more.
Given the fact that the data shows that a lot of these fish, once they're mature, don't necessarily return to the estuary right away, or maybe not at all, does that mean that a lot of the mature fish would not be susceptible to our traditional commercial and recreational fisheries that occur in our sounds and estuaries, which may provide a buffer for the stock against overfishing?
That was one of the hypotheses. I know Tyler Peacock, our PhD student working on this project, is really interested in looking at. It does look like that, you know, we do get some age 3 and 4 fish in the estuaries. A lot of those do tend to be male fish that look like they haven't gone offshore to spawn yet. So that seems like it's a possibility. What we seem to be learning from this project is that we are seeing them offshore in this mid-shelf habitat, possibly on artificial reefs. So I'd like to really have a better understanding of what those fish look like, what their age distribution is, to better understand that. But you know, I think what we're seeing is potentially consistent with that idea.
All right.
Okay, thank you.
Good. Thank you, Dr. Ash. All right, we're going to break for lunch now. So the next talk isn't till 1:30, but staff are going to be back outside at their posters and at their displays at 12:45. So please don't rush through lunch, but please come back and spend some time out talking to staff and maybe to some of the presenters and cover some more questions. But we'll be back at 1:20. Thanks. All right, so welcome back, and thanks everyone for coming back after lunch. So our next talk, the next two talks actually are going to be DMF staff. So first we have Amy Staples. She's a biologist. She works out of the Washington office, and she's responsible for our multi-species tagging program. And a little bit about Amy. She received a Bachelor's of Science in Fishery and Wildlife Management and a Master's in Forest Resources from the University of Georgia. And I think Amy and I started at the division about the same time and worked a lot together, you know, getting the tagging program going and stuff. And if you've ever caught a tag fish and called it in and received an award, you've probably talked to Amy or, you know, had some interaction with Amy. So today she's going to give a talk on tagging into science. So take it away.
All right, good afternoon, everybody. I see some people still filing in. Come on in. Well, I'm so excited to be here. I think maybe I've got the toughest time slot of the presentations because I got to keep y'all awake after lunch. So bear with me here. My name is Amy Staples. I am the tagging biologist for the North Carolina Division of Marine Fisheries. I'm based out of the Washington Regional Office, and I've been with the division since 2015. I've had many roles from temporary tagging biologist to statistician with License and Statistics section, and now I'm the full-time tagging biologist with our multi-species tagging program. And today I'll be talking to you about tagging into science. There we go. All right. Well, I can't start this presentation without talking about the multi-species tagging program. Raise your hands if you have a coastal recreational fishing license. All right, thank you so much for purchasing that license. The multi-species tagging program is funded through the CRUFL. So in part, you are doing your part to help us to fund some of the research that we are doing, like you've seen many of the research projects today here in this room. The multi-species tagging program was established in 2014. And we really established it because we wanted to standardize tagging research here at the division. There have been multiple tagging studies throughout time here. We started tagging striped bass in the 1970s, red drum in the 1980s, and so our biologists really wanted to make sure that we were standardizing that data collection. We were basically building a platform for if we wanted to have further tagging research, we could just build right upon it and we had everything in place, which included staff as well. So the current rendition of the program started in 2014, so a lot of the information that I talk about today will be from that time span. So what fish do we tag? So currently our program tags striped bass, southern flounder, red drum, spotted sea trout, and cobia. And for this presentation, I will be focusing on southern flounder. So let's start with what is citizen science. So citizen science is the collection analysis of data relating to the natural world by members of the general public, typically as a part of a collaborative project with professional scientists. So basically to say, citizen science is science for everyone. We want folks to get involved. That's what the tagging program is about. You've seen multiple researchers up here. We're collaborating with them. We're collaborating with recreational anglers. We're collaborating with commercial pound netters. We're collaborating together to do a scientific research study, and it takes all of us to do that. So with this talk, we want to talk about the opportunities that you have to get involved with the tagging program. So why do we tag fish? You've seen a lot of information in these studies today, so I hope you're seeing that we're kind of building layer upon layer upon layer. So each study is showing you a little bit more, and each thing that we do is a little part of the puzzle to southern flounder. So tagging studies play a vital role in assessing fish populations by providing information about migration patterns. Maybe we're looking at spawning patterns. Maybe we're looking at climate, seasonality. So it's giving us a little bit of information about those migration and immigration patterns. We're looking at habitat use. Why are those fish in that area? Is there something specific? Is it the habitat? Is it some type of water quality? What is it? Is it where the food is? And that food is there because of a certain reason, because of that habitat. We're looking at stock delineation. We've had several questions about that. We're trying to define that stock. Is it just a North Carolina stock, or is this stock for the South Atlantic, for the Atlantic states? Where are these flounder moving, and how do we manage them? We're looking at population structure. You know, we have a population that's full of young juveniles, or do we have mature females out there doing these spawning runs? And looking at growth rates. When we tag a fish, we measure that fish, we weigh it, and then when we release that, we're hoping that someone returns it and also takes some of that similar information so we can look at growth rates over time. And then additionally looking at mortality rates, that natural mortality and also fishing mortality from the different fishing sectors. So we've seen lots of presentations today, and they're all talking about the different tagging techniques out there, which include our conventional tagging, our acoustic tagging, and our satellite tagging. And these are all just different methodologies of tagging, but essentially what this is a mark and recapture study. What is a mark and recapture study? Well, we capture a fish using some type of gear. We then mark the fish, whether it's with a conventional tag, an acoustic tag, or a satellite tag. We release this fish back into the water. We don't want to tag dead fish, fish that are sick, anything like that. We want to tag healthy fish, and we want the tagging that occurs to not harm that fish as well, because we want those fish to survive so they can be caught again. And then, of course, we're hoping that that fish does get recaptured, whether it's by an angler, whether it's through a university researcher out there, or if it's an e-tag, getting that fish to swim by an acoustic receiver, or having a satellite tag pop off and send that data to a satellite. But this is a tagging study, it's mark recapture. So how can you, as a member of the public, university researcher, whoever you are, how can you get involved with the tagging program? Well, it's through these two ways. You can either volunteer and help tag fish, or you, as you're fishing out there, you can also recapture those fish and turn in those tagged fish. So how do you become a volunteer tagger? Well, step one, you can talk to me. Fantastic. I've actually talked to multiple people out today, so I'm so happy we've got so many people who are interested in getting involved. But what you'll need to do is you need to complete an online application. The QR code up here on your screen, that actually is takes you directly to the application on our website. Once you fill out the application, then you'll be added to an email listserv, and then I'll be able to email you when our upcoming trainings occur. You are required as a volunteer tagger to attend one of our trainings, and these are in-person trainings. We really want you to be there in person, not virtual, because we want to have hands-on participation. So at these trainings, they roughly run about 3 hours. We'll get an overview of the tagging program. Today, I am giving you the snack bar view of the tagging program. At the tagging program training, you're gonna get the buffet. You're gonna get the full meal. So we're gonna give you that overview of the tagging program. We're gonna review the proper tagging procedures and data collection procedures we have. I know a lot of people out there who actually tag for other states and they tag for federal agencies, but our tagging can be a little bit different. Our data collection can be a little bit different. So we want to go over that to make sure you fully understand the expectations that we have. Because our tagging program is not just, oh, we're gonna tag fish. We hold to research standards and scientific standards. So we wanna make sure that you understand what those are, and we'll go through that process. Then we're gonna have hands-on tagging demonstration and practice with real fish. That's right, I said real fish. They're gonna be there in person. Sorry, they're not gonna be lively, but they are there collected from our sampling trips. If you're lucky enough, maybe you'll get to tag one of the rubber flounder I have. You can go check it out on our table in the back over there. But also too, those fish demonstrations are gonna be done by the species leads, the biologists who are in charge of those fish. So you're gonna get one-on-one time with the biologist to pick their brains, ask them questions that you've been wanting to know. We want you to have that interaction with our staff. So definitely come to these, they're fantastic. And so once you go through an in-person training, you basically, if we have need in that area where you fish, then you're gonna get a tagging kit and you can start tagging. Sound good? All right, I got a lot of volunteers actually here today, and I was so excited to see so many people here, so I appreciate y'all coming and supporting us. So when are the next trainings? Well, the trainings are actually coming up in April. We generally hold trainings in spring and fall. So these are the upcoming trainings. The first one is occurring Thursday, April 11th in our Wilmington office. That's going to be 6 to 9 p.m. Then training 2 is going to be Thursday, April 18th. That's going to be located in Morehead City at our Central District office, again from 6 to 9 p.m. And then the next training is going to be Saturday, April 27th. That's going to be located in the Washington Regional office on Saturday from 9 a.m. to 12 p.m. And I will mention that training number 3 in Washington is a family-friendly event. I think fishing and tagging is for everybody, so I want you to get your kids involved. So if you've got kids 8 years and older and they want to come participate and learn how to tag, they can't get a tagging kit on their own, but they can see in the presentations, they can see how to tag fish, and you as the parent or guardian will actually be the owner of that tagging kit. We want the kids to get involved. We want kids to see the science that's going on, get them involved as early as we possibly can with management and interest in natural resources, and especially fisheries. So how do we tag Southern Flounder? I'm not going to go through the full procedure. If you want to see that, come back to the table and I can show you how we do that. But generally we tag Southern Flounder in the cooler months, September through May. We really try to avoid the heat of the summertime. That helps reduce the post-release mortality because of course it's hot out there. Fish are already stressed. If we start poking them and prodding them, we're only going to increase that chance of mortality. So we really avoid tagging during those summer months. We do tag the fish with a spaghetti tie loop tag. And so we tag fish that are a minimum 11 inches or more, and this is specific for our volunteer taggers. We tag with red and yellow tags can be double tagged. I'll mention a little bit more about that here soon. We do have specific tag numbers, so all of our tags that we put out, whether it's Southern Flounder, red drum, whatever, we have specific tag numbers for each of those tags, so we can track that fish wherever it may go. So, as I mentioned, Southern Flounder, we have single low reward, double low reward, and high reward tags. Those double low reward tags, that actually lets us look at tag retention rates. So if we get returns of those fish and maybe only one tag is in that fish, then now it lets us know, okay, are these tags the proper tags for that species? Do we need to modify our research any? And that also goes into our modeling. And then high reward tags, we have those. High reward tags are those $100 tags. That's right, $100 tags. And so that helps us look at reporting rates. So that's what the colors mean. North Carolina, we use yellow tags and we use red tags. Yellow tags are a low reward tags. You can get, if you were to recapture one of those fish, you can get a hat, towel, or fish pen, and red tags are worth $100. How many people have recaptured a fish out there? All right. I got a couple people out there. If you've ever recaptured one of those red tagged fish, you know that you have to cut off that tag and send it in. And usually we ask for your social security number because that's what North Carolina as a state requires. Well, as of this year, moving forward, we no longer have to get your social security number. We're actually moving forward with $100 prepaid cards. So that's a big win for us in making the processing of your tag returns quicker as well. So that literally leads into the second way that you can participate in the tagging program. It's by recapturing fish. So if you're not interested in tagging, because it's a lot of stuff, extra equipment in your boat, but you're out there fishing all the time, then definitely, if you're out there, check your fish, check both sides of your fish to see if it may have tags. And if you do have a tag, just remember to catch, cut, report. If you catch a tagged fish, cut off the tag and report the tag to the 1-800 number listed on that tag. There's also a website, that's the QR code for our online reporting form. So you can turn in that information online. Again, rewards for those tags include hats, fish pins, towels, and red tags are worth $100. As I mentioned, red tags do have to be cut off, and your tag will actually say that on there. It says cut off tag. The reason that we cut off tags is because if we have an issue with tracking that tag number, then we want to make sure you've got it in hand so that we can verify it. Red tags have to be mailed into the Division of Marine Fisheries so we can verify. Yes, people have tried to double dip, so that's the reason why we have you cut off the tags. So you're going to record information, not just the tag. We don't want to know just the tag number. We want to know information on that fish. And that information is the same. If you were a volunteer tagger collecting that data on the original tagging event, then it's the same information when you capture that fish. We want to know the date of the capture. We want to know location of the capture. And when I say location, I don't mean Neuse River. Doesn't help us when we're tracking the fish at all. And I will go ahead and tell you, I am not into giving away your fishing locations. I don't want to do that. So please, please be as specific as possible. If you can give coordinates, that is fantastic. It helps me track your fish better. If you can at least give us a city, a location, or point, that really, really helps. I don't want to give your data away. I just want to track these fish. Because again, we talked about what tagging research provides. Maybe it's a habitat use. Maybe it's a location. Maybe it's a primary nursery area. Maybe it's something we really need to look at and delve into the research even more or form a study over. So that's why we really want that specific location. So of course, then I want to know the tag number so I can actually track that fish through our system, the species. I want to know the gear used for capture. So were you out there gill netting or were you hook and lining? What's the total length of the fish? In some fish, we also like the fork length as well. And then the fate of fish. I want to know, did you keep that fish? You harvested it? Did you release that fish with the tag, or did you cut off the tag and then release that fish? I am not Marine Patrol. If you keep a fish out of season, you know, I gotcha. I just want the data. That's all I'm asking. I'm not Marine Patrol, okay? But let me know what you did. And if you do catch a tagged fish, I don't want it to change your behavior. So if you were planning on catching that spotted sea trout or that flounder during season and you wanted to keep that flounder, just because it's tagged doesn't mean you need to release it. If you plan to keep it, keep that fish and just report that tag. If you were planning on releasing it, release that fish. That is okay. Don't change your behavior just because it's tagged. In addition, when you report your fish, I want to know your contact information. I'm not going to spam you with a whole bunch of stuff. The reason that I want your contact information, if you provide your email, then you're going to get confirmation of your tag return with all that information. So you can look over it and let us know if that information is correct that you reported. Maybe we got your address wrong. Maybe the tag number could have been this or this. That'll let you verify your data. But also, I need your mailing address because I know you want that reward packet. And in that reward packet, we have a letter and certificate. That letter contains information with all the original information that we collected at the time of the original tagging event. The same with the certificate, however, the certificate has a map of the original tag location and the recapture location. So the more specific you are on your recapture location, the better your map is actually going to be in tracking your fish. And then, of course, you get to choose with yellow tags to get a program hat, fish towel, or fish pin, and then the red tags, again, are worth $100. All right, so let's talk a little bit of numbers here. How many Southern Flounder have we tagged so far? So this is just kind of a rundown of the species we've tagged from 2014 to 2022 in total, and then Southern Flounder, we've tagged over 8,000 Southern Flounder, and total recaptures are 551. Let's look at that a little bit. I know the maps are really small up here, but what I want you to gather from this is what we have literally been tagging from the Virginia border all the way down to the South Carolina border. So we are tagging all throughout North Carolina's coastal estuary waters, near shore waters. And you'll also notice, let me see if I can get the pointer going. I'll show you on both sides. These two little dots right here. Those are legit flounder tags. Of course, we've had different weather cycles, and some rivers get more saltier than others certain time of the years and from different years. And so through our electrofishing surveys, we were actually to tag flounder that far upriver. So those aren't, that's not an error, they were actually tagged there. So these are all the different releases that we have. And then from there, we've had 551 recaptures. And so just building on the data that we've seen from the other researchers, as you can see, we do have a southern movement that is occurring. And we do see those fish down in South Carolina, Georgia, and all the way down to Florida. Now, it's not been many, but we have gotten recaptures from there. And people ask me all the time, how do you know those were legit recaptures? Well, we call and we verify that. We ask them questions to make sure that that fish is one of our fish, that it is one of our tags, and that they actually caught it. We've had in the past folks call and say they're in Savannah, Georgia, and they got a tagged fish. when you start asking questions, you may have found out that they actually caught it from a seafood dealer, or bought it from a seafood dealer, and they found the tag when they were cooking it up. So we do make sure we're verifying that data to make sure it is a legit catch out there and not something that may have been caught in North Carolina waters and then transported some other way to other states. So some of the really cool information that I have here are the days at large and distance traveled. Now, days at large is going to be from the date from the tagging, original tagging event to when it was recaptured. And so we, on average, see about 141 days in between an original tagging event to when it was recaptured. But the maximum has actually been 4.8 years, and I'll show that fish here in a minute. And then distance traveled. Now, distance traveled is a little deceiving. We can't do things like satellite tags and acoustic tags where we can see that fish moving back and forth in through the waters. Distance traveled for us actually means from the point of the original tagging event to the point where it was recaptured. We don't, with conventional tags, we don't necessarily know that movement all the way in between. So it's from basically point A to point B, traveling by water. So the average distance traveled is going to be 16.6 miles. The maximum, though, is 518 miles. So let's take a look at these two specific fish. There we go. So most days at large. So this fish was out for 4.8 years. It was originally tagged in September of 2015 and then recaptured in June of 2020. And so this fish is really fascinating to me. It's only 8 miles in between the original tagging event and the recapture event. However, it's 4.8 years. We can see that growth rate. It grew 14.6 inches. So just from this data alone, we can guess that this was probably one of the larger females. And just imagine how many times that fish hopefully went offshore to spawn, came back potentially to that same area, went offshore again, came back, and did that over a 4-year period potentially. And that's what I'm saying is, what's interesting today is you have had the chance to listen to all these different researchers. So you can see it's not just one tagging study, it's all these different tagging studies coming together to learn more about these fish. And hopefully continued tagging studies, whether it's satellite, acoustic, or conventional tagging, or another tagging method that is still being developed right now. All this information is important and goes into the management of this species and us learning more about it. So the next fish that we have, this fish is the furthest distance traveled. So in 184 days, it traveled 518 miles from Hogpen Bay all the way down to Hobie Sound, Florida. Didn't grow that much, so we wouldn't expect it to grow that much during that time period, but it traveled that far all the way down to Florida. And it was from if you notice, the tag number is number 17, so it was a fish that we tagged really early on in the study. And again, just interesting to see these different movements of flounder. So what we've learned about southern flounder so far through all these different tagging studies? Well, we see that there's high spring and summer site fidelity, meaning that those early season tagged fish are recaptured in the same system prior to that fall migration. But those larger tagged fish in the fall are highly migratory. Those are the ones that are those spawning mature fish that are going to go offshore potentially or near shore to spawn. Then we have our general movements. So those larger mature fish are migrating to the ocean beginning late summer, early fall, and into the beginning of winter. And then a portion of the stock we do see is migrating south to South Carolina, Georgia, and Florida. There is evidence that the fish return to North Carolina estuaries after migration to the ocean, and we've seen that multiple times in the studies that were talked about today. There is limited tagging data, though, in South Carolina and Georgia, so there suggests a low probability of those adult fish moving north into North Carolina waters. Again, this is where more tagging studies can potentially help us tease some more of that data out. And then right now, that current assessment does combine Florida, Georgia, South Carolina, and North Carolina into one unit stock in the U.S. South Atlantic. So if you want to know more about the tagging program, please contact me. I am happy to take any questions or anything. Come back there and talk to me at the booth. And there is a third way that you can get involved with the tagging program, and that way is to also spread the word. You have learned so much today about tagging and tagging research. So let your fellow anglers know, your friends, your community know that there is tagged fish out there, and to look for those tagged fish. If they're interested in getting involved, have them sign up to be a volunteer tagger, or make sure if they catch a fish with a tag, don't hold on to that tag. Make sure you report it as soon as you recapture it, because it goes a long way in our data collection and our research and management. And with that, thank you so much for having me today. I appreciate it.
All right, thanks, Amy. Yeah, thanks, Amy. Good job as always. Do we have any questions? All right.
Maybe this is a dumb question, but do you have a preference? Like, would you prefer people to just report the tag, eat the fish, versus take the tag out of the fish?
Oh, that's a great question. We go back and forth on that all the time. One of the things when the tagging program study started is cell phones weren't in everybody's pockets. Nowadays, everybody's got a cell phone. Signals are getting better out there and stuff. And so there's been discussion about, how do we move forward? And it's not just our division, but other agency as well. Do we start putting, leave the tag on the fish? Or do we start putting QR codes on the tags for you to scan with your phone? So there's a lot of different ways that we can go. Right now, we want you to cut off that tag only because there's so many instances where the tag number gets reported wrong. And so if you cut off that tag and still have that physical tag in hand, we can verify that tag. Red tags, especially for sure, we want you to cut it off because we have to verify that tag number. So that's why we're kind of moving it that way. But we can also still track that fish. So some fish that you may catch with a conventional tag may actually have an acoustic tag in it, and you don't know it because you can't see it from the outside, but we know it. So maybe we're tracking that fish also through the acoustic tagging. We also use pit tagging, and then of course satellite tagging as well. So just because a tag is cut off doesn't mean we can't still track it. Even some of our conventional tags will have an internal anchor in it. So if that fish was recaptured later and harvested, you can still turn in that internal anchor tag for a reward.
Thank you.
Yeah. Question. Oh. Hold on 1 second, they're coming to you. I want everybody to hear.
How do you capture the fish for tagging?
I love that question, and I will say it depends. It depends on who is catching that fish. If you're a volunteer tagger, a lot of our recreational guides out there do catch our fish, and so it's a lot of hook and line tagging. And again, it also is species dependent as well. We have our independent sampling programs with the division, whether it's through our gillnet surveys, our sound survey trawling. We've got a lot of different ways that we sample those fish, even electrofishing as well. So really, we're not gear specific. The key thing is that gear does not harm that fish, and the fish that is tagged is a healthy, lively fish. And in our volunteer tagger trainings, you'll learn that we never want you to tag a fish that is sluggish, unhealthy, floating on the water. It's happened before. Don't tag a fish unless it is healthy and lively. So as long as that gear allows for that fish to stay healthy, then it's okay for us, and it's a legal gear as well.
Joe Luskovich again, East Carolina, and we use your tags on our fish. You heard the talk
Yes.
by Rebecca Ash. I'm wondering if you ever gave an idea or thought to having a lottery as a reward, like if you get a yellow tag, you get a hat or a pin or a towel, but maybe you get entered into, everybody who returns gets entered into a lottery and they could win $1,000. I'm interested in getting higher rates of tag returns
Yes.
from the conventional tags, and I'm trying to think how we could motivate people to.
Well, I'm glad that you suggested that, because actually that tells me we need to promote it a little bit better. We actually do have an end of the year drawing. So every tag that is entered or returned to North Carolina Division of Marine Fisheries, we have an end of the year drawing where 3 anglers from every species of fish that we tag get an extra $100 reward. So we do have that incentivized program. But I will say that internally we've been having those discussions because, as everyone knows, inflation has gone up, and it's also seeing that in the science as well. And so we've had discussions about that. Is, you know, our tag rewards and stuff have kind of been based on studies from, you know, the late 90s, early 2000s. Is it time for us to start upping that ante a little bit? And it really does somewhat come down to budgets of what we have as far as our grant availability and funding. So great question though, but that is some of the things that we're trying to work on is incentives. What type of incentives do we have, and can we make those better for anglers?
Okay, thank you very much for that answer. That's great that you're thinking about it. I just wanted to thank all the recreational anglers who did return tags, because we do use that data. We very much appreciate it. It complements the acoustic tagging. So thank you if you've returned a tag.
And really, you know, just echoing that, you know, a lot of people in this room have been helping out, whether you're division staff, whether you're a university researcher, a commercial fisherman, recreational angler. Everyone is taking part in this program, and that's why I call the tagging program everybody's program. It's the people's program. You know, get involved with it, and there's so many ways, and it really goes far. And it's active science. It's actively, you know, getting out there and participating. So if you're interested, please contact me. We'd love to have you help out. Whether you're just a student who's interested in wanting to learn more about tagging, please contact us. And I love doing outreach events, y'all. So if you have a classroom full of kids, if you have a fishing club, please call me up. I will travel and come talk to you about the tagging program. All right, thanks.
Yeah. All right. Thank you, Amy. So next, invite Dr. CJ Schlick up. She's our stock assessment scientist at the division. Little background on her. She holds a Bachelor's of Science from the University of West Florida in Marine Biology, a Master's of Science from Savannah State University in Marine Sciences, and there she did a snapper grouper acoustic tagging, I think, on Gray's Reef. Fun project, it sounds like. And then a PhD from George Mason University in environmental science and public policy, and she studied the population dynamics of river herring there. And before she was our stock assessment scientist, she also ran our age and growth lab. And she's giving us a talk today on the specific research needs of the Southern Flounder stock assessment model for the South Atlantic.
Thank you, Steve, and hello, everyone. My talk is not as lively as Amy's, but we will see what I can do. So today I'm discussing a lot of the research that I'm doing in regards to the Southern Flounder stock assessment. Sadly, I will not be presenting the 2024 update today. I am presenting the 2019 update, but I am going to present some of the information that goes along with it that we are looking into to better the model. So to give you a brief history, the Southern Flounder stock assessments were completed in 2005, 2009, 2014, and 2017. All of which concluded that the Southern Flounder was overfished and overfishing was occurring. The 2014 assessment actually was not used for management because it was due to us using a unit stock definition of only North Carolina. Tagging studies and genetic studies have shown that the unit stock should incorporate North Carolina through the east coast of Florida. So in 2017, a coordinated effort between North Carolina, South Carolina, Georgia, and Florida commenced to develop a stock assessment of the entire unit stock. This model successfully passed peer review in 2018 with a contingency that an update would be quickly done through 2017 because of expected changes in the recreational fishing estimates. The 2019 update is what I'm going to be talking about today, along with additional research recommendations that we have determined as needs while conducting the new update. The 2024 update is still ongoing, but I wanted to touch on several of the needs that we've seen from that. So as I said before, the 2018 benchmark stock assessment concluded the stock was overfished and experiencing overfishing. Low recruitment through the latter part of the time series resulted in a 100% probability that the stock was overfished and a 53% probability that overfishing was occurring in the terminal year. This was a peer-reviewed model accepted for use in management. The top figure that you see on the screen depicts the estimated spawning stock biomass in metric tons depicted with a gray line and the confidence intervals around those estimates in the dotted lines. The straight dashed line that you see across the top is the management target, and the solid line depicts the management threshold. When we are below the threshold, we use that as the definition of the stock being considered overfished. The bottom figure is the estimated mortality of the stock through time depicted as a solid gray line with the corresponding confidence intervals in the dotted lines. The straight dashed line is again the management target, and the solid line depicts the threshold. When we are above the threshold, the stock is considered experiencing overfishing. The 2019 update of this model concluded that there was a 100% probability that this stock was overfished and a 96% probability that overfishing was occurring. Again, the top figure is the spawning stock biomass with corresponding confidence intervals and management reference points. We are below the threshold through the entirety of the time series. The bottom figure is the estimated fishing mortality from the 2019 update with the corresponding confidence intervals and management reference points. And we are above the threshold through the entire time series except for in 2005. It was also evident from this landings data that a larger proportion of the catch were immature individuals, and recruitment was still low and continuing to decline. However, the focus of my talk today is not the gloom picture from the previous assessments. I'd like to discuss the assessment itself and some of the research that we need to examine to provide the best scientific information for management. So getting into some of the specifics, for this model, we are using the Age-Structured Assessment Program, or ASAP model, as a preferred selection. The unit stock is defined from North Carolina through the east coast of Florida. The starting year is 1989 because this was the earliest year for which the shrimp trawl bycatch estimates could be generated, and the terminal year was 2017 because it was the most recent year that data were available for the 2019 update. We have an assumed birth date of January 1. Sexes are combined in the model due to a lack of information about the sex in the catches. But we use female-specific weights at age for the spawning stock biomass calculation since females grow significantly larger than males. We use ages 1 through 4 in the model, where age 4 is a plus group. Southern Flounder can live up to 9 years of age, but very few individuals are observed in the catch or in our independent studies over the age of 4. So we group those older individuals into that 4+ group. So anytime I mention age 4 from here on out, it will be a 4+ group. This model was run on with age-specific natural mortality, and maturity is based on previous studies that assume 100% maturity by age 4. There are 3 fleets in the model that represent the commercial fishery, the recreational fishery, and the shrimp trawl bycatch. The commercial fleet includes harvest from gears such as gill nets, pound nets, gigs, and trawls, and the different catches from each state are combined into one commercial fleet. Also, discards are combined into one commercial fleet as well. The recreational fleet includes harvest from gears such as gig and hook and line, and we combine all of the states into one recreational fleet with discards as well. Discards are not modeled separately. And the commercial shrimp trawl fleet also includes all dead bycatch. So here are the total landings, including discards, from North Carolina to the east coast of Florida for each year in the ongoing assessment. The landings and data from each fleet are represented in metric tons. The commercial fisheries seen in green and the shrimp trawl bycatch fisheries seen in the dark blue at the top of each bar have both declined over the length of the time series. However, the recreational fisheries seen in light blue has increased since 1989. The Southern Flounder fishery in the South Atlantic has switched from predominantly a commercial fishery to a predominantly recreational fishery. A total of 8 fishery independent surveys were used in the stock assessment, 3 recruitment surveys, and 5 adult surveys. The recruitment surveys measure the trends in the number of juveniles joining or recruiting to the population, which are represented as age 1 individuals in this model. The surveys are from North Carolina, South Carolina, and Florida. The adult surveys measure the trends in the number of adults in the population with one survey from each of the 4 states, and an additional survey, which is the CMAP trawl survey, to represent the ocean proportion of the population. So looking at those surveys, here are the 3 recruitment surveys that were done that are measuring the abundance of age 0 fish. Now, all of these are targeting age 0 fish in the population. However, recruitment is a process where fish are being recruited or available to the fishery. So recruitment in this scenario is age 1. For each of these surveys, the data point is bumped by 1 year and 1 age to become an age 1 survey. For example, North Carolina relative abundance of age 0 fish in 1989 is roughly 2.45. For the model, the relative abundance of age 1 fish in 1990 is that 2.45 value. All 3 recruitment surveys show a general decline overall in the course of the time series. However, I do want to note that Florida's decline that we are seeing here is actually due to 3 kind of outliers. And when you're statistically looking at this figure, it really does not have a trend throughout the time series. So I lump Florida's into a no trend index. The 4 adult state surveys used in the model vary throughout the time series. However, North Carolina, South Carolina, and Georgia all demonstrate a general decline throughout the entire time series. South Carolina shows some increase in the last few years, but it's still down overall. Florida is the only survey without a decline, but it also doesn't show an increase either. It's just high variability through time. Ooh, wrong way. So the last index we used is the CMAP trawl survey. This is another adult survey. We used the fall data from the inshore strata of the CMAP trawl survey, as this is when the adults are captured in high regularity to actually track the abundance through time. However, several strata were not sampled in the fall from 2018 through 2022 due to COVID restrictions and budgetary cuts. So these strata had to be removed when they had an incomplete time series, which resulted in all of Florida strata and 5 of the 6 North Carolina strata being removed from this index. Those are highlighted, the yellow highlights you see in that table on there. Those are just the years with the missing data. So the CMAP index with truncated strata has a relative abundance without overall trend. So remember, recruitment in this model is when juveniles are available to the fishery and is assumed to occur around age 1. Age 1 recruitment, represented as the green line on this with the blue dotted lines as the confidence intervals, represented in thousands of fish, has declined from roughly 14 million fish in 1989 to roughly 4 million fish in 2017. The female spawning stock biomass is the total weight of all sexually mature females in the population. It's also depicted in green with the confidence intervals in the blue. And it's represented as metric tons. It has shown a decline in the last 10 years of the stock assessment. The terminal year of 2017 had a spawning stock biomass that was almost half of the initial estimates in 1989. The model population had a decreasing number of Southern Flounder, but also we wanted to note that it is dominated by age 1 and age 2 fish. In the figure you see on your right, or on the right side of the screen, all of the black are age 1, all of the purple are age 2. The blues are your older fish. Most of the stock, or as previously stated, very few fish are found older than age 4. Less than 1% of males and less than 4% of females reach age 4 or older. And these were estimates directly from the model. However, these were informed from our age sampling. So what you see here is I've plotted the proportion of each age of all the samples collected by all states where an age could be ascertained from the otolith. These samples are from independent and dependent fisheries sources, and the age data were used to inform the model of that previous figure. We see the same trend with very few fish reaching age 4 or older. And when we examine the samples through time, these percentages of older fish have actually decreased. 5% of the samples from 1999 to 2010 were 4 or older, and then the next 12 years, in 2011 to 2022, only 2% were 4 or older. So we are seeing a decline in the older fish. So I said I was going to talk about some of the research recommendations that could help improve the stock assessment of the species. There's a long list of items that many people in this room have touched on today. However, I'm going to focus on two areas that are very specific to stock assessments: the indices that inform the models and the selectivities estimated in the model for each fleet. So the indices are the fishery independent data sources used to provide the model with the trends in abundance. Having indices with the two different trends that we have seen are problematic for any model, especially when all of the indices are weighted equally. Additionally, the indices are to represent the stock as a whole, not just localized patterns of abundance. So I'm examining appropriate ways to combine these indices within the model to represent the overall stock abundance. The selectivity refers to the ability of the fishing gear to catch certain sections of the fish population, usually in reference to size or age. Selectivity is an area that needs continuous research with any model because this could be a huge area of uncertainty, but it plays a critical role in the information about the indices in the fleets. Exploring temporal and spatial differences within a stock can provide needed information about how selectivity should be treated within the model. So, getting into what looks like a really crazy graph. For the indices, I decided to perform a sensitivity analysis looking at each of the data sources for the indices independently. On these graphs, the red line is the base model, so what is actually reported in the stock assessment report that we give everyone out. Each other color is where I have stripped all of the additional indices out except for only that data source. So the Florida model only has the recruitment index and the adult index from Florida. All of the others have been stripped out. I would like to note that the only Georgia and the only CMAP do not have recruitment surveys in them. They are only adult surveys. And as you can see from this figure, when you do this type of analysis, you can come up with varying different trends within the spawning stock biomass and the fishing mortality over the course of the entire time series. And this could indicate that including all of the indices evenly weighted within the model creates confusion, for lack of a better term. So I wanted to combine the indices based on the trends the indices were exhibiting. You have the base model in red, and then you have the non-changing trend in blue, and that is the Florida and the CMAP indices. So the CMAP trawl, the Florida adult trawl, and the Florida recruitment surveys. And then the green line are those indices with the declining trends, the North Carolina, South Carolina, and Georgia surveys. And the model is sensitive to which pattern in the indices is used, particularly within the early years of the model. However, all three scenarios had kind of the same conclusion in that terminal year of the model, where they came to very close ending points. Not to jump away from that, but there is more information to look into for the selectivities that I wanted to talk about today too. Models can be extremely sensitive to the selectivity of a fishery, and the problems in selectivity can result in biased estimates and relate to unrealistic estimates of catchability. The benchmark model estimated selectivity using a logistic regression for the recreational fleet and a double logistic regression for the commercial fleet. These regressions are based on a relationship between 4 age classes that we used in the model. However, a logistic regression requires 2 parameters, a double logistic requires 4 parameters. This can create some mathematical problems within a model when you only have 4 age classes. So I did a sensitivity analysis where all of the catch selectivities were estimated for each age. I used the same initial values that were done from those logistic regressions, and then allowed the model to vary those initial estimates based on what was best fit for the model. The SSB on the left-hand side and the fishing mortality represented on the right-hand side, the SSB, or spawning stock biomass, increased when we use these selectivities at age, and the fishing mortality decreased when we use these. I do want to note that this was an initial run where the fit of the model was not the best. The base model was still a better fit. However, the model did converge. But we do see a major change. And I could continue looking at those initial starting values to see if there was an even better place to start. But I saw that where things were not fitting right, they did not follow the same pattern through time. And I believe a lot of that has to do with the fact that selectivities can change over time with current model, which our current model configuration cannot take into account. With management actions, fish selected for by the commercial and recreational fleets can change based in size and composition. Length and age frequencies change through time, and these selectivities need to be addressed in the model using selectivity time blocks. One critical need for research is how to appropriately assign these time blocks to each given fishery. And to give you an example, I have only plotted the North Carolina commercial harvest on the left and the recreational harvest on the right with the changes in management that could impact these selectivities. Changing the size limit can impact your size selectivity. Changing a bag limit can change your retention within the selectivities. So all of these have to be looked at as well. Now, I have only provided the management actions in North Carolina. We also have numerous management actions within the other 3 states as well. So along with the changes in time, we need to account for differences spatially. The unit stock of Southern Flounder is managed independently in each state, thus the selectivity impacted by management strategies differs from each state. One approach I am researching to account for this is using a fleets' area approach, where each state's commercial and recreational fleet are modeled independent catches within. This does create a more complex model that requires a lot of data and more computational abilities that have not been available a decade ago. But we need more information as well on recruitment, movement and dispersal, and rich data sources about the abundances within each of these areas, as well as how all of these relate to each other throughout the unit stock. But with appropriate testing and research, it should provide a more robust model overall. So a stock assessment determines the health of a stock that is undergoing fishing pressure, as well as understanding that fishing pressure. To fully grasp the utility of a stock assessment, we need to research the impacts of management on the assessment. The stock assessment created in 2017 was a good model that provided a relevant baseline to take management action with. However, we still need to adapt assessments to the management put into place given how these actions change the fishery and thus impact the model directly. Further studies into selectivity and the indices used are needed to ensure the best scientific information is available for management of the Southern Flounder stock. And while this presentation focused on researching parameters within the stock assessment model, the model itself cannot be constructed without the research presented by the other authors at the symposium, as well as other research that occurs throughout the species range. The model needs information from the life history, catch and bycatch rates, stock genetics, growth rates, age and length information, maturity schedules, mortality, and much, much more. Through these collaborative efforts, researching the population dynamics and modeling our impacts on those dynamics helps to develop a greater understanding of the valuable resource. So there's no way I can thank everyone that has worked on these assessments, but I've started a list, and I appreciate your time and your attention today. If there's any time left, I'll answer any questions.
All right, thanks, CJ. Good talk. Do we have any questions?
There's one over here.
What factors can you speculate on that would affect recruitment?
There are a host of them, from just environmental pressures to climate change to prey availability. You know, there is a stock recruitment relationship within this model, so it's very loose, but you know, a lack of females could be not producing enough recruits. But I do think there's more environmental variables that are really impacting this species.
given the more recent estimates of 50% maturity schedules, will you be doing sensitivity runs with those new estimates in your 2024 update?
I am actually doing sensitivity analyses with that. I set one to run Monday evening and just have not been able to open the computer to look at it. The model does take some time to actually process through, so we would love for it to be fast, but computers haven't caught up that much.
How long does it take for one model iteration?
If I run it through a full Bayesian analysis, it takes 14 hours. So, pray you don't make a typo.
Yeah. All right, way at the back. Run, Charles.
These guys are saying I used all my questions up, is that right?
No more.
So, I might have missed this, but I'm trying to better understand the stock assessment schedule and aligning that stock assessment schedule with management. So right now we're using a stock assessment from 2019 with terminal year 2017. Max age for the species is what, 7 or 8? And so we're using a stock assessment to manage in 2024, and those fish from that assessment aren't even in the water anymore. And so I'm trying to better understand, how do we fix that moving forward? Can you talk about that some?
I can a little bit. I presented the 2019 today. The 2024 update is actually using data through the terminal year of 2022. We are trying to finish that report and get it out to everyone. I'm adding the last few additional sensitivity analyses to it, and trying to get it out later this year, probably in the fall. But part of the issue with the timeline of a stock assessment is when we say 2022 is the terminal year of the data, we have a lag from when the data are done being collected December 31st to when they're actually available for analysis. And that lag right now is about 6 months. So it's 6 months before the data ever come into my office. And then there are a lot of quality control checks that we look through, make sure everything's good with the data. For instance, with the 2022 or 2024 update, I put out the request for all of the data. It was ready the first week of July. I didn't actually have it ready to be put into a model until the last week of September. So it just takes that long to process that much data. And then when you start running the models, if everything looks perfect, you can have a model run within 2 to 3 weeks. Models usually don't ever look perfect. So if you have to do any kind of diagnostics in the model, every diagnostics can add a week to your timeline. Like we said, the model itself has to run for 14 hours uninterrupted. And that's every time I run a different analysis. So the base runs that you saw where there were 8 different runs on the model, that took 8 days to do. So it does take time to research what could be impacting the model, and making sure that we are putting in the best scientific information into all of it.
And I appreciate that question, and I'm going to add a little to it. I'm going to put my administrator hat on. So we have 3 stock assessment positions at the Division of Marine Fisheries. Right now, CJ is our only stock assessment scientist. And, you know, we recognize that this delay in stock assessments, you know, affects management decisions. You know, a lot of times we're forced to bring these results to the commission for policy decisions and acknowledge that, you know, the terminal year may have been 3, 4, sometimes even 5 years, you know, prior. We are exploring other avenues to look to maybe contract some of that work out. In the interim, we've had a very difficult time recruiting and retaining stock assessment scientists at the state. A lot of it's got to do with it's just a very, very small niche. I mean, it's a very quantitative heavy profession, and there's just not a lot of y'all out there. But yes, that's something that we recognize, and we have conversations about it every day. We would love to improve the timeline in between stock assessments updates. But, you know, right now, I can't force CJ to work 80 hours a week, because then I might lose her. So, but thank you for the question. So.
Better computer would always help.
Yeah, we'll work on that. All right, well, if there's not any more questions for CJ, thank you. Good job. So right now, we're going to take a short break. Yep. So right now, we're going to take a short break, let everyone have an opportunity to refresh their beverages, maybe run to the restroom, and we'll have John Carmichael come up at 2:45 to deliver his keynote address. All right, so next is our keynote talk. Mr. John Carmichael, who's the Executive Director of the South Atlantic Council, is going to give us a talk on the common questions and concerns about marine fisheries management and science. And before I turn it over to him, I just want to give a little background on John and his pedigree and why we asked him to give this keynote address today. So John's worked at the South Atlantic Council since 2003, and there he was the program manager for the CDAR program, and that's the Southeast Data Assessment and Review Program, and that's the program that conducts all of the stock assessments for the Southeast and the Gulf region. Prior to that, he worked for us as a stock assessment scientist, and I think Director Rawls mentioned this morning that he worked on river herring and striped bass, and, you know, two species that, you know, are never contentious or anything like that. But, you know, he's one of the few people that I, myself, and, you know, a lot of people have said that, you know, he can take some of the most complex topics and distill it down to, you know, its essential parts and convey it in a way that, you know, everyone can understand. He's also been involved in the management world, the fishery management world at the federal level for a long time, and he's got, you have 30 years of experience? I'll say you got 30. You look like you have 30 years of experience. In bridging science and management and, you know, advising managers on how to interpret science, what science can tell you, what the limitations of that science is, to inform fishery management policy. So with that, I'm going to bring John up, and please open up your minds for some wisdom.
Hope I can do a good job with the remote as the last two speakers did. Not go back to us old timers from the morning that struggled. But yeah, I really appreciate y'all that stuck around till after lunch in the afternoon to hear this. Really a pleasure to come here and talk about this. You know, I'm certainly not a flounder expert, wouldn't pretend to do so. You know, worked in the division. I didn't work on the flounder assessments. I did work on striped bass, river herring, red drum, things like that. But, you know, one of the things that I'm really passionate about is communicating scientists to managers and fishermen. And I like to think of it as kind of taking some of the fear out of stock assessments, because a lot of times once you say stock assessment, people want to leave the room because they're like, man, it's going to be a bunch of figures and science and I'm not going to understand, and equations. But, you know, I was kind of lucky to study under Dr. Joe Hightower at NC State for my master's, who's an excellent teacher in stock assessment and really great communicator. And being more of a biologist than a mathematician or statistician, you know, the way he taught it was really from the biological perspective, and I've been able to turn that into really sharing that with fishermen and managers throughout my career. And, you know, because ultimately we are talking about living creatures and they're following particular rules that all populations follow. There can be a lot of complicated science and a lot of complicated equations, but you don't have to know all that to understand what's going on with the population. So let's see if I can make the slide work. All right. So what I'm going to go through here is what I say is like a top 10 list of questions that I've heard through my career about stock assessments and science and fisheries management. So first off, what is a stock assessment? And it can mean a lot of things. To some people and to a lot of laymen and fishermen, they think of a stock assessment as basically this. There's some biologist and he's just throwing darts at the dartboard, and there's either no fish or there's so fish you can walk on them or whatever, you know, that just really don't understand it, so it's a mystery. So you think this is how it is. There is a sort of official definition that those in the biz think of, and it's about using statistical and mathematical calculations to quantitatively, so that's the important part, characterize the status of a stock, predict the response to management choices. So the key things are quantitative. So you just saw the presentation on the stock assessment, and you see the numbers, not just that it's overfishing, but this is how much it's overfishing. So you're not just giving things in a qualitative sense, but you're able to put values on it, which then managers can use to figure out what to do, how do they respond. The other important part of it is predicting what's going to happen in the future. Because as we just mentioned on the Southern Flounder assessment, you know, the assessment is a number of years old, and we're trying to manage today, so we had to project and predict what the population will do and respond to different management things. And that's a really important part of it. So it's not enough to just know what's going on in the past to really have an effective assessment. You've got to predict how your management and such is going to do in the future. What it all comes down to visually is kind of numerically explaining this. So you have a stock, that's the dolphin there in the middle. You have what adds to that stock in the green, where you have recruits, which we mentioned, that's the new fish being born. We'll refer to them as a year class, or I'm going to use the word cohort. That's all the fish born in a particular year. But you know, the only way you get new fish in a population is they're born. And the only way you get more mass in the population is those fish grow. So you have inputs and recruits and growth. And then you have removals from the population. Natural causes, we call that natural mortality. And then whatever surplus that keeps the population stable and you can take out, you can remove from fishing mortality. So if all of these things are equal, then you have a nice stable population. If you've got more coming out than going in, you've got a declining population. And so the model that you just saw has lots of equations and takes lots of different data sets and data sources to try and put numerical values on all of those arrows that are going into and out of that population. But biologically, these are the basic factors that are at work. And this is all an assessment is doing. The model is keeping track of the different ages of fish and their maturity and their size, and just a big bookkeeping experience really within the actual numbers to explain all this within the rules that govern populations, which are the same rules that govern any population, whether it's bugs or people or fish or deer. There's certain rules that all populations work under, and that's what the fisheries models are using. And what the assessment ultimately is doing for us is telling us answers to some key questions. Is the removable rate sustainable? Are we taking the right amount of fish out? Is the population sustainable, and can it go through those bad years? Because the reality is we're not going to always get good recruitment years. Some years are going to be good, some years are going to be bad. We've got to leave enough fish out there so when we have bad years, I've got enough spawners that I can get enough to keep my population going. And I need to go through however many bad years may occur. Also importantly is what can we expect for the future? And often in stock assessments, people are really interested when you first present it to a group of fishermen. Usually they want to know, what's the fishing mortality rate? And am I overfishing or not? But often that's the history. That may not even be all that important. What really matters is what's the future? And I always tell fishermen, forget about what the fishing mortality was in the terminal year, which as CJ just told you could be a couple of years back. That's water under the bridge. Think about what you can fish next year, because that's when the regulations are going in. So you really focus on the projections. And me as a scientist, the first thing I want to look at in a stock assessment is, what are the projections telling me? How does the projected catch that I can take, say, next year or the year after compare to what I'm taking today? Because regardless of what the status is, that's what we as a fishermen are feeling. What's the difference between what's predicted and what I'm actually taking today? And then importantly, and we've heard a lot about this today, what factors might be impacting the population that aren't just fishing? And, we've seen environment, we've seen habitat, we've heard about climate change, we've heard about struggles with, what's going on with spawning and where fish are going. So how is all that impacting the population? And, it's often pretty easy to say, well, if there's these other factors, I should be able to keep fishing. But the reality is, if you go by that, then eventually the population's going to be gone. There's not going to be any to catch. So the unfortunate reality to fishermen is if there are other factors that are impacting the population, it often means that, yes, indeed, you do have to catch less. If there's excessive development and loss of habitat, then that's going to reduce the inputs to that population, and that leaves less to come out for fishermen, unfortunately. And that's one of the toughest things that there is to deal with, is that, it's not always fishing that's the cause of a population in decline, but because fishing takes a surplus, you do have to share in the cost. Great question I get all the time. Where do you get your data? And it's almost always delivered like that. Where do you get your data? Well, the simple answer is from you, fishermen. I participate in a thing called Marine Resource Education Program. They train fishermen. Almost always get that question. Where do you get your data? Well, you heard about all the data sources in the assessment. You saw the information on tagging. We depend on fishermen to give us most of our data. In the fisheries science world, we break it out into two main types: fisheries dependent and fisheries independent. So as they sound, fisheries dependent is what we get from you, the fishermen. Fisheries independent is what we get from the other people in the room, the university researchers, the division researchers, the scientists. That stuff that's done independent of the fishing. That's all of the studies into migrations and behavior and age and growth, the indices that are so critically important. But a big part of this is the fishery dependent. And I'm going to talk some about how that factors into the assessment and why that's so important to us, particularly understanding how many fish are in the ocean. We can get a good handle on mortality and such just from fishery independent data. But if we're really going to put things into perspective of a population in the real world, in the real ocean, impacted by real fishermen, we've got to have accurate fishery dependent data. And one of the reasons is because fisheries assessments are all about counting kills. Most people think of natural resources management as the managers are out there in the world and they're looking at how big a population is. Let's say it's trees in the Croatan forest. The foresters go out there and see how many trees there are and then tell the loggers how many they can cut. Fisheries isn't like that. Fisheries does it in a whole different way. We don't know how many fish are really in the ocean until everyone has caught them, because we add up the kills that happen over time. So it's more like in fisheries that we're at the mill counting the loads of logs coming in, and then we're figuring out from how many trees came off a piece of land, how much timber was in that land. So the fisheries dependent data is basically the fishermen telling us what they did. And then it allows us to look back over time and say, okay, this is how many fish were in the population back then. And that's a really fundamental difference from how most people think natural resources operates and how the world of fisheries science truly operates. And there are some ways that let us get a better idea maybe of what's out there in the ocean and some survey methods. But for the most part, most fisheries still relies upon this business of counting kills back in time to recreate the population and know what was there. And in doing that, lots of data sources are considered. We've heard a lot about them today. You saw a bunch of them just a minute ago presented, from the commercial sampling, the recreational sampling, understanding population structure, understanding spawning behavior. All of that stuff is super important to telling me what those arrows on that first figure with that dolphin are doing in this population, and how each of those arrows is interacting with each other to give me a population that's going up, going down, or staying stable. This is a great question. I always love this one. Why should I tell you about my catch? Won't you just use it against me and close the fishery? I follow the OBX board on Facebook because I like to see what's fishing. I love to go out there and go surf fishing. Recently, there was a post about blue line tilefish seasons, and somebody literally posted, you shouldn't be telling them surveyors what you're catching. They're just going to shut it down on you. People still have this attitude, and the truth is, it's really not that simple. As I said, we rely upon fishermen for data. They're going to come up and ask you what you caught. People will say things like, yeah, right, I don't trust MREP. I'm not telling you what I caught. You're going to shut the fishery down. The reality is it's not that simple. And hopefully from the presentations here today, you've seen that there's a lot of data sources that go in there. There's a lot going in to the stock assessment. And the reality is taking part in the data collection is important, and being accurate is important, because if the scientists have good numbers, they're trying to do the best they can with them. I haven't really run across any scientists in my experience that are out there trying to game the system to keep fishing down. They're trying to put the most accurate and unbiased model together that they can to give you a good picture of what's going on in the stock. So the fisheries data, dependent data, are just critical. And the reality is it's impossible to game the system. I've told people, it's like, if you can figure out how to lie to a sampler today to influence an assessment that's going to be done 5 years from now, you got a lot better input on what this model's doing than I do. because I have no idea how all the pieces are going to play together. Best thing you can do is tell the truth. And the other thing is these analyses depend on many years. So the assessment goes back to 1989. So bad data from 1990 is in there. Somebody that misled a port sampler in 1992, it's in there, and it's in there forever, and nobody ever knows it, and all of us today pay the price for the fact that people may have been intentionally trying to mislead. And you don't know that that happened, so it's in there. The other thing is more data almost always reduces uncertainty. Uncertainty is just, how good a handle do we have on these answers? And the more people participate in sampling, the more truthful they are, the better information we have, and the less uncertainty we have. And the reality is, with all those other data sources out there, if fishermen collectively decided to try and mislead on what they're saying they reported, it would be caught up with through the age sampling, or the decline in the population over time, or with the surveys, or maybe like a tagging program. It would come out. It may take time, but eventually there'd be some, what we modelers would say, lack of fit. What one data source is telling me is not the same as what another data source is telling me, so things aren't fitting well. You eventually get to the bottom of it and you say, oh, there's just more error in that data set. So the less error we have in data set, better models we have. Another issue is that those forces that demand regulations, like Fishery Reform Act here in North Carolina, they don't make exceptions for bad data. I work in the federal system. We're under the Magnuson Act. The Magnuson Act makes no allowance for bad data. We in the South Atlantic have to manage knobbed porgy with the same level of detail and precision expected that the New England Council manages Georges Bank cod. Vastly different fisheries, magnitude, sampling, and value. But there's no exception for that. So bad data is not an excuse to not manage. So getting good data is critical. And then finally, reporting can be mandatory. Commercial fishermen, y'all are well aware of this, for-hire fishermen that are in the federal system, you're aware of that now. And I even hear there's chances that it's going to happen in North Carolina with recreational fishermen on some species. We're going to say you need to report, you need to get your data. So can't say enough about how important fishery-dependent data are to us. And then tags, I love the tagging study. Participated in a lot of tagging study over my time. Everyone loves tags, right? You think people are putting in those tags data. You just saw all the great information that can come from a tagging program. I was at a North Carolina Northern Advisory Panel meeting 25 years ago talking about a striped bass assessment and trying to use tags. And there's a guy in there who's like, I got a coffee can full of tags on my boat and I'm not going to turn them in. And why not? You're going to use them against me. You're going to think those fish died. I said, well, I got news for you, buddy. I already think those fish died. He's like, what do you mean? I said, a tagging model assumes every fish is dead. The only thing that you're telling me really in terms of the modeling that I'm going to do is how long that fish lived. I said, so you may be sitting on a tag that shows me some striped bass was at large with that tag in him for 10 years. But I don't know it because you haven't turned in that tag to me. And we talked in about high reward tags. We were looking at a $250 high reward tag for dolphin. Would that be enough to get people to turn in any tags? And asking these guys about a high reward tag. And I asked the guy, I said, if I gave you $50 for that striper tag, would you give it in? No. $100? No. I said, $200. His wife was at the meeting. She's like, $200, I'm bringing them all to you tomorrow. So, it's like, what is it worth them to give the tag? But the fishermen had this idea that if they turned in those tags, then we're going to say, yep, that fish died. Because they think we have this estimate of the population that's independent and alive, and we're subtracting their catch from it, and if the catch is too much, we're going, oh, you caught too much. But as I said, we're not. We're counting the kills. We've got to have accurate kills to do an accurate count. And then the other thing is just look at all that neat information we've seen today that come from tagging programs. It's pretty rare even today to get fishing mortality estimates out of tags. It's a great way. It's a great tool. It's independent of all of the things you saw in the stock assessment. It's independent of knowing accurate catch and aging fish and doing surveys. But it's so hard to get enough tags coming in. That program about 5% tag return overall. You need 10% or better to really start getting somewhere in terms of getting a good mortality estimate. But with enough people tagging, enough people committed to turning in the tags, maybe we could get there. And it's great if you can have two measures of fishing mortality for a stock. One coming from all the infrastructure of catch and age and those models, and another coming from something that's actually pretty straightforward and simple like tagging. But it's just hard in the ocean. There's a lot of places for fish to go, a lot of ways for them to die. This one is good. What's the big deal with ages? Heard a lot about ages. You've heard a lot of things today. Talking about aging, I think maybe Fred started out showing some otolith. Here's a picture of an otolith. we determine the age of a fish looking at various bony structures. And then it's important for us to understand the age composition of what it is that you catch. And that's a really important input to the stock assessment. We're assigning fish to what we call cohorts, which I said is all the fish born in a particular year. So I'm going to get a little bit of science here. We're going to kind of, move through it. I'll throw out a little bit at a time, not to be too intimidating, but this right here is one of the most critical parts of a stock assessment. If you can understand this, and I'll explain it to you, you can really understand what's going on in a stock assessment. This is a picture of catch at age. I think this is actually a river herring catch at age cohort from many, many years ago. Try to use some stuff that's relevant. They're out in the ocean. They don't start coming back into spawn until they're 2, 3, 4. So what you get often in a catch at age picture is some catch a young fish, and then it increases to some particular age. In this case, it's the age 4 there that's the peak. That's what we call the age of fish that are fully recruited to the fishery. So all age 4 fish, if they're encountered by the gear, they're susceptible, they can be caught, people want to catch them, they're legal. All the things that influence whether or not you keep a particular fish. Well, after that point, you see there's fewer 5s and fewer 6 and 7s, 8, 9s, and so on. We call that in fisheries the descending limb. That is the impact of mortality. That's fishing mortality and natural mortality. So think about it for a minute. There's more babies alive today than there are 50-year-old people. And the babies that are born this year, there's more of them alive today than there are going to be when the 2024 babies are 50 in 2074. This is a basic rule of populations. There's more young than old because things die over time. That's simple, basic, we understand it, right? And then, depending on what your mortality rates, you may die faster. So the people that smoke cigarettes don't reach as maximum age as people who the people who smoke cigarettes don't live as long as people who don't smoke cigarettes. They have a higher mortality rate. You would see it in a figure like this for humans. So what the stock assessment is doing is essentially just looking at that, what we call the descending limb from age 4 to, in this case, 9+, and saying, what's the slope of that? How fast did these fish decline over time? It's just a slope. It's just like if you cut a rafter or built a set of stairs and did a stringer, rise over run, that's all this is doing. The ultimate equation is just the decline. It's just a slope. And that's why ultimately the underlying math is fairly simple. People worked this stuff all out with a piece of paper. It wasn't as complicated as it is today, but this is the fundamental thing. If we know how fast the group of fish is declining over time, then we know how fast they're dying. And then I can compare that to how fast I think they should be dying and know whether or not I've got a sustainable population or not. And the model is just keeping track of this for every year, every cohort that's in the model. That's where the complicated equations and bookkeeping come in. But if you can start to understand this, and I'm going to talk about it some more to try and reinforce it, it's really the fundamental thing that's going on in a stock assessment. Another great question is, are those surveys any good? Scientists don't know how to catch fish. Can you tell you how many times I've heard about that? Scientists don't know how to catch fish. You guys go to the wrong place, you go at the wrong time, you're using the wrong gear. Well, yeah, that's exactly right, because they're not like this guy putting every hook in the water they possibly can to make sure they come home with something. They're trying to fish different. They're trying to fish in a consistent manner. And they do this because the most important thing from a survey is that changes in the survey catch are due to changes in the fish population, not due to changes in fishermen's ability to catch fish, not due to them going where the fish are at the particular time, etc. They need to cover all the places where a fish are and where a fish could be. And there's been a lot about that, like, oh, those guys are fishing in the wrong place, and then a real push sometimes to say, okay, can we make our surveys better by fishing in, more and more of the fish are. But now we're talking about climate change and we're worried about species shifting. That wasn't a concern when I was in grad school and learning about fisheries. We weren't worried about climate change. We weren't that far from worrying about the next ice age. We certainly weren't worried about global warming because we didn't understand what climate change really was about and the importance of variability. But now as fish are shifting, it's becoming even more important that we have samples from a wide area to see where fish could be today that maybe they weren't in the past. So that's what scientists are doing. They're not trying to catch the most fish. They're trying to be consistent. That way, they can determine trends in the fishery that they see that may not agree with trends in the landings based on fishermen. And that enables them to do things like know how effective regulations are. If the landings go down because of a regulation, you would expect that the survey maybe will stay stable or go up because the population's doing better. But if the scientists in their surveys were chasing after fish as well, you wouldn't see that. So it'd be much more difficult. So they're just doing it in a whole different way for a whole different reason. And, you know, some of them can catch fish. The other thing is the surveys tell us about the rest of the population. So we heard this morning about understanding the larvae and where the spawning fish go, and then the juveniles and the larvae and coming back into the sounds. There's a whole bunch going on. I mean, look, we can see from flounder, there's things potentially happening to adult flounder out in the ocean that we're not capturing in any of our fishery dependent data. We've got to get that from fishery independent data because we've got to understand the whole population. Now, I'll bring back my catch at age picture, and so remember, like, this is what's caught, but we know that there's a population out there that's more ones, more twos, and more threes. And this is the other thing the survey does. It fills us information in about how many ones, how many twos, how many threes are there. And it's really important to know that because if I'm not getting fish born, if I'm not seeing recruits, if I'm not seeing recruitment to the fishery, where's the breakdown happening? Do I not have spawners? Do I not have egg survival? Do I not have larval survival? Do I not have juvenile survival? I mean, I'm sitting there today thinking about that whole spawning cycle of these flounder. And, you know, there's a lot of places for things to go wrong. They got to go out in the ocean. They got to find each other. They got to get their eggs fertilized. They got to come back into the sound. So, you know, the surveys allow us to understand the whole population and figure out where those bottlenecks are occurring and what may be the impediments to this population that aren't just related to fishing. Because if the impediments are not fishing, it's really unlikely most of the time that we solve the problem simply with fishing. We do have too many examples of, you know, even reducing fishing mortality, not resulting in improvements to a population because we really didn't understand what the problem was. And one of my long-term concerns is as we learn more about warming waters and shifting distributions that we're going to face more troubles like that in the future. So it's really important that we have these good surveys that cover everything. This one is a great question, lots of fun. How do you know how many fish are in the sea right now? And this is one that people definitely like to throw at me as a stock assessment guy. Whenever you start talking about something, how can you know it? You know, the ocean is a big place. There's a lot going on out there, and it really isn't a matter of just using the magic 8-ball or the voodoo talisman or flipping a coin. It is, you know, it's using those models and seeing what they can tell us about a stock of fish. Challenge is, how many fish are out there? You know, if you ride down the forest, you can tell when they cut the trees down. I was riding around Carteret County this weekend, and I noticed a lot of places that used to have trees, and now they're just dead hulks because I guess seawater rise. You can see the loss of trees. You know, you can see the loss of terrestrial things. But I could look out across the waters and not know if there's any more fish in there than there were 20 years ago. You know, that's the challenge. This environment is foreign to us. So we have to go through the efforts with these models to try and understand, you know, how many fish are here? And it's tough. I never sugarcoat this. It's tough to know the absolute abundance of fish, a marine fish in particular, in the ocean. We do that by focusing on that cohort, understanding the descending limb. That lets us get mortality rates well. And we can estimate mortality rate for a population from just a sample of the catch. I don't need to have the absolute catch to know what the fishing mortality rate is. And in fisheries for many years, that was the overarching goal, was to just estimate the fishing mortality rates and adjust management accordingly, because knowing that we didn't have a good handle on the overall catch. Because we need that overall catch if we're going to scale things to the actual population size. That's critical. In the federal system, we manage under MSY. I think the FRA requires a lot of similar things. You know, maximum sustainable yield, that's expected to be the absolute for the overall population. That requires some biomass scaling that's supposed to be reflective of the scale of the population. And that's really, really difficult because that requires that catch information that we count on fishermen for, and it needs to be accurate. But it's not just the catch. I also need to know all the fish that are being killed. So I need to have accurate information on discards. And as hard as it is to get accurate on catch, it's even harder to get accurate information on discards, but it's required. And it also needs selectivity, and you heard a bit about selectivity in the assessment presentation. That's just vulnerability by age. So I need to know, you know, you caught X number of 4s. Well, can you land every 4 you catch, or is it just a fraction of them? because that's going to be important to scaling this stuff up again to the whole population. So we do the best we can with that, but where the models really shine is with the relative measures. So looking at fishing mortality, we call it F in the fisheries world. It's just, the mortality that's caused by catch relative to whatever that rate is that gives us the maximum. So you'll see a lot of assessments, and I think it was in the figures that showed the status, F over FMSY or F over some reference point. That's just the relative level. And that's usually really well estimated, even if I don't have a good handle of the total catch and struggle to get that overall abundance. The same with the biomass. You look at the biomass relative to some reference level, usually the biomass that produces the maximum sustainable yield. We love that word. I didn't really define it. It's just, it's expected to be the average yield that a population can produce over time. It's the highest average yield it can produce. Requires us to know a lot of stuff that's tough. Again, absolute scale, it requires us to know recruitment, and that's hard to get at. But these relative measures are very robust. So we can adjust management based on relative measures that might say you need to cut fishing mortality 25%. Well, we can take management and try to reduce catches 25% and see how it works out. And for a lot of years, that's really all fisheries management tried to do. And in some cases, it worked quite well. The other important thing about this is recall, and we heard the question there at the end from Mike about, hey, the assessments from 2017 to 2022, working on 2024, what's going on? Assessments tell us about the past. That's all they can tell us. The model tells us about the past. The projections that I mentioned, those are where we learn about the future. And knowing next year is a lot harder than knowing about the past. This figure came out when the South Atlantic was first working on red snapper a number of years ago. The fishermen are going, it's like this, and they said the scientists are going like this. Well, both were right. The scientist had an assessment that was a couple years old. These crazy fish produced the best year class they'd produced in 25 years with only about 2% of their maximum spawners out there. There were virtually no red snapper out there left to spawn, and they produced an incredible year class because the spawning potential of fish is simply incredible. I mean, coastal striped bass recovered early in a moratorium, not from fish saved in a moratorium. They recovered from a handful of fish that had survived mortality, excessive mortality for 20 years. Most fish populations do that. North Carolina red drum recovered, had a great year class, coincidentally came along about the time the first management plan went in. Kick-started that population in a great way. So this can really happen, that pass, but when you get that thing, you get this perception that, scientists don't know what they're talking about. Well, their data was just a little bit out of date, and it's hard to keep up with what's going on right now. We need to know that. We need to know what's going on in the future. And if we're going to tell you how many fish are in the ocean tomorrow, then I need to be able to predict the future births. I need to know how fishermen's behavior might change. I don't know what the environment's doing. All those things are tough. What we do know, important thing, if we reduce fishing mortality, populations tend to improve. That's pretty obvious. The calculations are going to make that happen. The hard part is, how do we really reduce fishing mortality? Because even when we reduce regulations, fishermen are often really good at continuing to catch fish despite increasing regulations. Because fishermen are good at catching fish, that's the bottom line. So I'm going to sneak in a little more science here on you. I mentioned cohorts, I showed you the figure. In the model, you're going to get it as a table like this. And I want to point out a couple things because it does tie into that last question from the stock assessment. So I mentioned this is the fish born in a particular year. So the red numbers going down in the diagonal, the big bold red numbers, that's the 1981 cohort. They were born in 1981. They were 1 in 82. They were 2 in 83, and so on. They're gone by age 6 in 1987. So if we added up that catch backwards in time for those red numbers, that would tell us there at least had to be that many fish alive. This is the crux of the stock assessment. There were at least that many fish in the population because this is how many that you caught that were born in that year. Make a slight adjustment for those that die from natural causes, and there's your stock assessment. The important thing about this is you notice in the early years, over to the upper right, some of those years, those cohorts, I don't know much about. That 1976 cohort, I only ever observed them once when they were age 6. I don't know much about them. I don't know much about 75. I only observed them 2 years when they were age 5 and age 6. Well, that's the past when I was worried about that, but think about the future. I come down to 1993. That's my terminal year of my assessment that we talked about. I'm doing this assessment in 1994 with 1993 data. I'm going to take management in 1995. So now you see how the years are progressing. I don't know really much about the recruits in 1993. I've only seen them once. Those fish, which one does the pointer? Right in here in this area is where the trouble occurs, right? See, I've only seen these fish once. But think about this going forward, 1 year, 2 year, 3 year, 4 years. Pretty much then all of the fish that are dominating the population 4 or 5 years from now in my projections, I didn't know much about. This is why the timing of assessments becomes so critical. This is the issue we're talking about, right at the end there. And this is why it's so important to do assessments on a regular basis, because we're really good at knowing the past, but it's much harder for us to know the future. And it's just because of the nature of this, because I said, we're counting kills. We're not trying to estimate how many fish. We don't have a way to say how many fish for this population, say, we're going to be there in 94 and then subtract your 94 catch. We got to depend on counting this stuff backwards in time. So getting a little bit off the hardcore science now, how can the stock be in trouble when the fish is great in my area? Fishermen say this a lot. They really perceive this, First thing is, it's a really big ocean out there. Well, I said, a fisherman can be an expert in his neighborhood, but a lot of times, if you took a wall chart that covered the Atlantic coast, he'd be well served to be an expert in an area you could cover with your thumb. Maybe a commercial guy that's out there every day with your hand. But a lot of times, we're dealing with stocks that are covering a much broader area. Here with flounder, we're looking at North Carolina to Florida. No one fisherman is an expert in what's going on with this population over this entire area. So sometimes what you're seeing is not what somebody else is seeing in another area. And the other thing, of course, is you could just be a great fisherman. There are fishermen who are able to catch fish and catch fish consistently, even if populations are declining. That's just how it is, And I know flounder. I've never been much of a flounder fisherman. But, flounder fishermen are a different breed. Those guys that focus on flounder and catch flounder religiously, populations go down, they're going to keep catching them. And it's the same with most of our fisheries. It's the same with commercial fisheries. Our snapper grouper complex has 55 species, but there are guys who are specialists. We got one guy who's responsible for, a number of years ago, well over half of all the vermilion snapper, because that's what he catches. Talked to him recently, he tried to catch some black sea bass, said he couldn't catch a black sea bass. The other guys around him catching black sea bass, he can't catch a black sea bass, but there's a vermilion in the neighborhood, this guy could catch it. Because people just get sort of specialties. So, they may not perceive the same reductions that the overall stock is in other areas are perceiving as well. The other thing is a more scientific word, what we call hyperstability. So what this means is catches can stay strong while the population declines. Because fishermen, they're always fishing to catch more fish. They're always getting better. You're getting better gear, better electronics. You're trading intel. There's a whole social media world now for recreational fishermen to give you all the tips. Tackle shops are filled with things to make you catch more fish. What that means is, as the population goes from being big to a little smaller to smaller, this red circle of what's being caught stays the same. That's hyperstability. You don't perceive the decline in the population from the information from the catch. And it's because the big word we fishery scientists use is catchability. Your ability to catch fish is going up. Technology is a huge thing. People's ability to catch fish today is much greater than it was 20, 30 years ago. Better electronics, better boats, better motors, braided lines, the intel that you have from communications, the navigation systems, spot lock trolling motors, all those things are having a huge impact on fishermen's ability to catch fish. So the population around you can be declining and fishermen continue to catch. So yeah, how can you tell me the population's in trouble? Well, those scientists are surveying all of this. And these guys up here, they're no longer catching anymore wherever they were. So their survey is showing that, well, the population isn't what maybe you think it is. The other thing is range contraction. Most populations, wild populations, have kind of a core range. And as that population gets declined from whatever causes, it tends to shrink down and fill in this core range. So you start with a healthy stock that's covering this whole big green circle, and as the population reduces from whatever reasons, you end up in the, sort of this red, it's core area. So people in that core area, they may have never seen any decline in the population. In the council, when we first started dealing with those red snapper and that funny cartoon picture I have, Folks off of North Florida were like, what are you talking about? We've never seen a problem because that's the core area for red snapper. People in North Carolina hadn't seen a red snapper for 20 years. Just they weren't a part of the fishery. They weren't that abundant, particularly up off the northern coast, you know. But as that population expanded and started filling out what, you know, say the green area, then suddenly they start seeing that fish again. So if you're in a core area for a fish, then you may not see the same decline that somebody else does. Again, that's why it's important to know about the fishery over its whole range and to have those surveys that are covering the whole area. So the stock is rebuilt. Get there with flounder, hopefully. Why are the regulations so strict? This is another one as well. People have this idea, fishermen often have this idea that, you know, population's not really rebuilt till I go back to those regulations. I had a guy tell me on striped bass something like, you know, till I can go back to keeping 10 fish at 12 inches, it's not rebuilt. Well, sorry to break this to you, but you're never going to be there again. And that's just a harsh reality. And, you know, fishermen often think, wow, you know, population's better, I should go back to those regulations. But the reality is in fisheries is those regulations created a problem in many cases. They were too liberal initially. And here's just the breakdown of the flounder regulations, the recreational, over time. You know, it went from 13 size and no bag to 15-inch size, one bag in a 2-week season. It's a very common progression. And what happens is population starts to get in trouble for whatever reasons. You have regulations that aren't doing anything. No bag limit, 13-inch size, that's not really doing much anything. So you start to be concerned. Managers are concerned. They raise the size limit an inch. People don't like it, but the reality is these fish grow fairly quick. So maybe you protected fish for 6 months, but then you're just catching them again a little bit later. So that's a very short-term impact on the population, doesn't do much anything. The bag went from nothing to 8. That may not have done anything. And there lies another big difference between fishermen and managers when it approaches the regulations. Then you go from, you know, a 15-inch size at 8 to a 15-inch size at 4. Well, when you cut the bag from 8 to 4 to fishermen, that's a 50% cut. Well, if when the bag was 8, most fishermen were only catching 2, the managers would look at that and say, that's hardly any cut at all. In fact, that may not be having any impact. Most cases, when you see bag limits change like that, you're only often having a very slight impact on the actual catches. Because what really matters is what's the average fisherman catching against what the regulation's going to be. But in almost all instances, the debate about possession limits comes down to what it is and what it's going to, and that's the cut. But what really matters to the population and to the managers is what's your average fisherman catching out there? What's your catch per trip on average? If your catch per trip on average is 3 fish and we lower the bag to 4, we had no impact on the fishery. As scientists, we tend to advise managers, you're gonna get beat up for going from 8 to 4 or 8 to 3 or 8 to 2. So if you're gonna get beat up, do something that makes a difference. You know, if most people are catching 3, you should make it 2, because you're gonna get the same beating for going from 8 to 2 as you are from 8 to 4 for the most part. So, you know, the regulations are then gonna have to stay low because your population is more abundant. Think about it. The reason they weren't catching the 8 fish is because the fish weren't there. So when the population recovers, the fish are there. They're easier to catch. Suddenly everybody can catch a flounder. A guy like me that doesn't, you know, just fishes opportunistically can catch a flounder. And many management plans go that route. It's not the guy who was able to catch 8 fish when they were rare that's going to put us over the limits and lead to the increase in harvest for the most part, because those guys are few. It's the average fisherman who wasn't catching any that suddenly starts to catch some. So when our populations become more abundant, they're more available to fishermen, more of the fishermen are catching them, well then your catch rates go up and more fish are being harvested for a given regulation. So the reality is, at the end of a rebuilding time, you've got a much bigger population out there and you're gonna have restrictive regulations to keep from getting into the same problems that you have that led to the issue. And that is one of the just most difficult things to, you know, to discuss with fishermen and to deal with management changes, that, you know, you're not going to go back to those regulations, unfortunately, because the goal was more fish. More fish is easier to catch in almost all instances. So all of this, why bother? Management. Populations are cyclical anyway. Hear about cycles all the time. And biggest problem there is you can't rely on cycles. Seldom do we know the cycles. Mother Nature has a way of changing the cycles just when you think you understand the cycles. And all of that was true even before we started talking about this latest situation of warming waters and rising sea levels and what's going on out there in the ocean. So here's a pretty typical progression of a fish population and how cycles get interpreted. You know, you start out here and we have this period where things are going up and down. Well, it got pretty low there, but you know, it turned around. And then it turned around. And in here, I'm kind of on it where I was here on average. So it's just natural variation. Populations rise, populations to fall. There's really nothing to worry about. There's no fishing problem. And then you get something like this, where you get an increase for a couple years and there's great optimism. Things are wonderful. Look, see, we didn't need management. This population's doing fine. Well, then things start slipping. Even when you get to this point, even right here, it doesn't look that bad relative to the long term, even though we're in a pretty significant decline. So it's like, you know, the cycle's going to come around. You know, the North Atlantic Oscillation will catch up. We'll be El Nino or what have it. We've heard them all. You know, it'll turn around. It'll be okay, but maybe not. This is real data. This is the black sea bass population. If you haven't seen that presentation lately, this is South Atlantic sea bass. This is where we are. We went into a decline. It's lasted 12 years for bad recruitment. It's not cycles that are going on. And it's very dangerous to fall in cycles because you can quickly find yourself waiting for that turn. It's going to turn next year. It's going to turn next year. The next thing you know, well, it didn't. And with rebuilding fish, chasing those cycles and chasing every little blip and, you know, a little bit better recruitment puts you in rebuilding for an eternity. The summer flounder stock, starting in the late 90s, was in rebuilding for like 20 years because every time there was a slight increase in recruitment, they caught more fish. And they argued about catching more fish. They're pretty much one of the reasons the Magnuson Act went to 10-year rebuilding schedules and 50% chance of success, because there was arguments to go after the fish every time it got a little bit better. And so rebuilding went on forever. You know, you've got to let things play out, and you can't really trust these cycles because they really never have worked out in any fish populations that I've seen. And so, are they cyclical? Well, yeah. Populations are not stable. No fish population is as stable as the projections that we have for, you know, rebuilding maybe 20 years into the future. They do go up and down. They zig and they zag because it's, you know, we call this field that I work in population dynamics. Dynamic just means things change. So we're studying the changes in populations. We expect change. So yeah, they're not stable, but can we as humans understand their cycles? We don't know. And even if we could from the past, like I said, with what we see going on in the environment today, it's quite likely that the future is disrupting any cycles we might have thought we understood in the last few decades. And the other reality is, whatever the population is doing, if fishing doesn't adapt to the downward trend in a population, you can easily push a stock too far until it can't recover. You know, it's like there's a certain buffering ability in a population, and it can absorb changes, but at some point, you go too far, and it's not going to recover. It's like a bad storm filling up a lake. You know, that dam can hold back that water and hold back that water until suddenly it's over the top, and then it can't hold anything. So the people downstream thought, oh, it's great, it's great, it's great, right? You know, it's raining real hard, but look, the river's not rising. Well, once the lake filled up and the dam busted, it rose really quick. And unfortunately, that's what will happen a lot of times with fish populations. Well, things fine, hyperstability, everything's going great, till it can't. Then we got a low stock and we hit a number of years of bad temperatures or bad rainfall or whatever really else is driving a stock, and they just don't recover. You get below the critical mass, and you may be a really long time before you actually get your stock to recover. And then the real factor is humans, you know. We disrupt every natural cycle we touch. That's just the nature of us being on this earth. So we really can't trust cycles to get us out of fisheries holes and to avoid taking the medicine that goes with fisheries management. And then the last question here for me is, why does my state have stricter regulations than those around me? And I think this one is, you know, really relevant to flounder, seeing that, you know, North Carolina is leading the charge, has the Fishery Reform Act. You know, I live in South Carolina. South Carolina's approach to flounder has been make some changes in the regulations and hope to stock flounder, actually raise license prices to be able to stock flounder as a way to saying, yeah, you know, we're helping out North Carolina and we're helping out the region with this problem. So, you know, North Carolina is left with far stricter regulations than other places. So there can be a lot of reasons for having stricter regulations. There's biological. Where is the state in the species range? If you're the state that has the core range, then likely you're going to have stricter regulations to protect that stock. If your state's on the fringe of a stock's range and you only see that stock a little bit of the year or only once in a while when conditions are right, your regulations probably don't really matter that much. You know, what Virginia does with Southern Flounder probably doesn't mean anything, really, to the overall stock. Then there's the fishery. How much effort is in the state? If a state has a lot of effort and a lot of fishermen, then you're likely going to have more stricter regulations because you've got more people going after the fish. Then there's political. And politics is there. We can't get away from it. Is the state interested in the stock? Is it something they care about? Is it something that their fishermen care about? And they say, yeah, there's a problem. Do we want to do something about it or not? Because, you know, in the state waters, a lot of times the regulations are set by legislatures. You know, North Carolina, again, is a bit of an exception. Out of my state of South Carolina, the legislature decides the rules. They might decide, oh, we're not that worried about that fish, so we're not going to change those rules. Politics really plays a role, particularly in the state-managed fish. And then there's a management reality. So is there some kind of regional authority? And that is one way to overcome differing state politics on a regional fish and can actually require all of the states in a range, potentially, to have some sort of regulations. And, you know, the fish don't care about the state borders, you know, so we're all talking about states, and we're about state regulations, but I think as we saw in the studies, these flounder don't care. Flounder go from here down to Florida, have a good time. What's that Florida catch doing to the North Carolina recovery efforts? That's a big question. So we do have these regional management entities. As I said, you know, I'm with the South Atlantic Council, the federal entity. But, you know, we do the federal species. State species are done by the Atlantic States Marine Fisheries Commission regionally, and they cover Florida to New York, deals with the anadromous fish and the state water species. In fact, most people in the room are well familiar with them. It's kind of a love-hate often, you know. I don't know if it comes up as much as it did when I worked at DMF, but there used to always be proposals going around the commission about getting out of ASMFC, and we're not going to do what those guys tell us anymore. We're not going to have that bunch of Yankees coming down here and telling us how to manage our fish. But, you know, the regional bodies can be really good when you have a fish that is migratory or multiple states are interacting on them to make sure that there's some parity and folks are doing their part to help, you know, sustain the fish and to help rebuild the fish as the case may be. You know, and then in North Carolina, you guys, as I said, are unique with the Fisheries Reform Act. It requires action. Can't just let the fish go. It does require something to be done to rebuild the population and protect it. So, you know, regional management, sometimes it seems like the good thing, sometimes it seems like the bad thing. You know, you really have to judge it whether or not other states are willing to jump in and help you on a new stock. ASMFC manages a lot of stocks. Would they be willing to take on another stock? You know, I don't know. That's a question for other people. And are other states willing to get involved in it? You don't know. But, you know, if you do start to become concerned about harvest of, you know, spawning fish, if they are going sort of south and east when they go out, could be something that could have some value for the stock, or maybe the other states will just collectively go along with it. But it is unique on the Atlantic to have ASMFC with the ability to enforce management regulations. So a lot of people don't know is there's also a Gulf States and a Pacific States Commission. The Pacific States Commission mainly involved in data collection. The Gulf States is like the Atlantic States was up until striped bass in the 80s, in that they write management plans, but none of the states have to follow them. So they do plans for all of their species, and they recommend management regulations, but nobody has to do it, and there's no consequences if they don't do it. What happened with Atlantic states was the concerns about striped bass in the 80s and said, we've all got to work together. So they passed the Striped Bass Act, which forced all the states to manage striped bass together and have consistent regulations. That worked pretty well, led to passing the ACFICMA, they call it, which is Atlantic Coastal Fisheries Conservation and Management Act, and that gave the ASMFC authority to require states to actually implement the regulations in the management plans that they develop. And all the states sit at ASMFC, they have boards that make the decision, state reps sit on the boards, pass things, and then the states are supposed to go back and do it. What the act does is it says if the states don't do what they said they were going to do, they can be found out of compliance, and then the Secretary of Commerce can shut down their fisheries. So there's real teeth to make sure the states comply with things that come in state waters out of Atlantic states, which is a pretty unique situation, but it has been very effective, and well, I guess effective we can argue because, any of our success stories have a tendency to turn the corner on us. But, it certainly made a difference in striped bass for a while, and maybe that population's facing other challenges today. But, overall, it does at least give the states a place to come together and talk about how to manage things. So, let's see. Yep, so thank you guys for coming, sticking around till the end, listening to this. I know it's late in the day, and I'd be glad to have any questions that you might have.
Well, I appreciate that, John. Wow. That was a good job.
Thank you.
I had a staff member actually text me during that and say, they did their college, courses in fish and wildlife and has worked here for many years, and that 1 hour, they said captured, the essence of the struggles that we have and, the job that we have to do best in anything he's ever heard. So thank you. Before I turn it over to questions, I do have one question. So,
uh-oh.
No, it's easy.
Yeah.
So, us here at the Division of Marine Fisheries, under, the State Statute Fishery Reform Act, we're tasked with collecting the data and doing the science and advising our Marine Fisheries Commission, who are the policymakers, on, that science and, what's going on with the stock, and based off the information that we have, what's, an appropriate course of action. And, I didn't really get a good sense for this until I worked with you at the South Atlantic Council and sat on that council as, a representative of the state, and, was faced with those decisions where, I had this, advice, this scientific advice with an uncertainty associated with it. And then I had this management decision, and there was this uncertainty associated with the potential management actions. And, we were tasked with making decisions that incorporated that scientific uncertainty and that management uncertainty that, was a real effect on people, was a real effect on industry. And, I remember, I think it was my second council meeting where we actually had to make a decision on, I think it was Deepwater group, or I think it's when we had the, Deepwater complex, and, it was up in Kitty Hawk.
Yeah.
It was a miserable discussion, but I would just love to hear, just your thoughts on how we, at the division and, any of the scientific, community can, better communicate our science and communicate the uncertainty surrounding those results in a way that makes sense, or a way that is realistic in the management conversation.
I mean, it is tough. Communicating science is hard. Like I started out, when you say stock assessment, even the biologists want to leave the room. And it's just such an intimidating topic. And the math that's underneath of it can be tough. And like CJ says, 14 hours running this model, you're like, how can I know what's going on with that, And it's funny, when I first started with the division, we had one of those all, big meetings, everybody was there. And I talked about stock assessments, and Lynn Henry comes up to me, longtime biologist in Northern District. I know Cat worked with him. He's just like, I don't believe none of that stuff you do. You stock assessment guys, that's all smoke and mirrors, And I was like, stick around, Lynn. Hopefully I can, after a while, convince you that it's not that bad. It's not that far removed from, the biology that you know so well. And, I've always thought when you communicate this stuff, you've got to start with, the assessment models and the fits to parameters is all well and great for the scientists that live in that world, but it's of no use to managers and fishermen. And you have to show it to managers and fishermen in a way that's currency that they understand. It's about, the abundance, just simple abundance of the populations, about recruitment trends, tying things back to surveys, to the catch statistics, the stuff that an average person can look at and see, okay, I can see what's going on with this population. You really have to strive to, you know, distill it down to its basics, to, you know, and present it in a way that says, you know, these are really your choices as a manager, because the uncertainty sometimes can be daunting, and it's easy for someone that wants to push a discussion or influence a decision to grasp at little details and throw them out there, you know, and not look at the big picture. But fisheries, you've got to look at that big picture of the population, is the way I've always tried to present it, you know. I hate to look at projections into the future without looking at the assessment of the past, because you may have not any idea, you know, there's no perspective when you look at projections of, say, abundance without looking at the trend of past abundance. It's useless. And virtually no stock assessment's really presented in this way. And, you know, one of the things I've always done is, you know, there's a set of figures you do for peer review when I was a functioning stock assessment scientist, and then there's a set of figures you do for managers and decision makers. And I was at a Northern Division presenting the striped bass assessment one year. They talked all night. They said, you got 5 minutes. Give us your assessment presentation. And they're like, you know, you're not going to do it. I was like, all right. I showed them one figure, you know, abundance, landings, recruitment trends. Here you go. I'll give you one figure. We'll talk about the assessment. You know, and I feel like you have to be able to give it to managers in that simple way, because that's all the attention span that Terry Pratt had to listen to me that night anyway, you know. And he thought he tripped me up. So you have to really, you know, make sure people are prepared to say, you're going before the managers, it's a different world. You got to present this information, and you've got to show them, like, what do you really know, and be honest about what you know and what you don't know. I've always, you know, approached this as being, you know, excruciatingly honest with the managers about what we really know. Had a situation with mackerel years ago. You know, we had assessments of MSY at 8 million pounds, assessments of MSY at 12 million pounds, and they bounce around. And, you know, one of the council members was like, are you confident that it's 8 million pounds now? And I'm like, well, I'm as confident as 8 as I was that we thought it was 12 3 years ago. But I'm pretty darn confident that it's 10, because it's sort of where it's always been, and it's where the fishermen always said it was. And that, you know, diffuse that situation. You have to really, you know, the other thing is, you know, as a biologist or a manager, you have to really understand what the sort of perception is out there and be prepared to deal with it, you know, and to face it head on and not say, oh man, I hope they don't ask me this question. My thought was always, what questions are they going to answer me? So I have the answer in my back pocket. And so, you know, you know, as a scientist who, say, presenting an assessment or a management proposal, you're the expert. You know about it in and out, and it's really, you know, I always tell my people, it's your responsibility to then distill that down to a way that the fishermen and the managers can understand it. Not to make them think you're really smart because all the big words you used and all the great figures you showed, but, you know, you have to communicate it or you're not really relevant.
Thank you for that,
Yep.
John. Appreciate that. All right, do we have any questions for John in the audience?
Come on, Mike. Mike and I go way back, so I give him a hard time a little bit.
Sure. We actually came from the same advisor, so.
That's right.
You know, one of the things we struggle with as a stakeholder in this process, it kind of gets to the question that Steve was asking, is in light of uncertainty, consistently the precautionary principle is applied. And so as, you know, you've got a lot of experience here, John. What are some of the ideas in which, when faced with uncertainty, the direction doesn't always have to only be less catch, especially if the assessments demonstrate that it wouldn't even make logical sense to be more precautionary in the face of such uncertainty?
No, I mean, that's a really good point. And the federal system that I operate in really doubles down on that, this idea of precautionary principle. And what that says is, if you're uncertain, you catch less. And what fishermen over 15 years of this have realized is that whatever's going on, the consequence is almost always catching less. But the reality is uncertainty is usually a two-tailed situation, as we would say statistically. I've got an average, and you know it could be higher or it could be lower. What precautionary would say in terms of the catch is that, okay, it could be higher, it could be lower. You've always got to go lower. And there is a lot of frustration amongst fishermen that, you know, at every iteration, you apply that, and then you end up with a catch that can be seemingly much lower than it needs to be. And it's, for me at least in the council, it is so ingrained in the federal law that it's very little that we can often do to overcome it. It's one reason why, you know, I think systems like ASMSC with more flexibility have a bit of advantage. You know, in that in some cases when a stock seems to be doing well and we have a better sense of maybe there is some bias to this uncertainty, do we need to be as strict and lower the catches as much as we are trying to do? Because sometimes lowering catches can have, you know, other consequences that you weren't prepared for. Deep water fish, you know, if you just try to lower catches but you don't really try to effectively manage effort, you end up with discard problems. Or you put in a size limit that leads to discard problems of fish that die anyway. So, you know, you've really got to have a good handle on that overall picture and try to do the best management that you can, but you can have politically oriented factors like the precautionary principle that do prevent you from, and sometimes, taking advantage of obvious abundance. And we also have a scientific system that is, you know, really strongly conservative in many cases, which tends to say, you know, well, you know, I'm not quite sure. Maybe we should not catch those fish yet. Let's wait another year. There's a human nature tendency really to not make decisions in the face of uncertainty. And the uncertainty doesn't even have to be relevant to the situation that you're making a decision about. And there's some elegant psychological experiments that have been done that I've read about where, you know, when faced with an uncertainty of some sort on one part of their life, people are hesitant to make decisions about other parts of their life. And that plays out, I think, in fisheries a lot of times where, you know, managers can be a little slower to make a decision, maybe to make the common sense decision because there's uncertainty. And I think that works against us. You know, fisheries, most of the time, my mantra has always been, if we can do pretty good management today is better than great management next year. And if you think about the time, the passing, the year classes, I mentioned the North Carolina Red Drum Plan, that may not have been the amendment everybody wanted, but it was something that came in with some restrictions and some caps on the harvest at a time when the stock came along and gave us a good year class and really kick-started that thing. You know, maybe it wasn't perfect management at the time, but it was timely management and it helped save a situation. And fisheries almost always responds to either being quick, doing something pretty good quick, or in really hitting it with a sledgehammer. So you think of those ratcheting down bag limits and slightly increasing size limits on flounder, it's no wonder that that didn't really do much, you know, because fishermen are quickly able to overcome and adapt, because that's what they're really good at, catching fish. And they're able to overcome and adapt slight tweaks to management so you don't have really impact. You've got to hit fisheries with a sledgehammer before you really see any response because of all that uncertainty.
John, I want to thank you for putting up the one slide where you show how a species or a population kind of draws towards the center as it declines. And here in North Carolina, we hear it all the time where, you know, geez, there's so many fish out there, you can walk on them, you know. This year we can go out there and catch tons of them. Yeah, that's true, but perhaps that's exactly what's going on. And, you know, range-wide, the population is declining. We might be the center of that population, and there are still a lot here. And so I wish DMF would show that graph more. I think it's very explanatory, and thank you for putting that there. Thank you, Roger.
Are you concerned that an increase in regulations could lead to like a decrease in the amount of people that purchase licenses? I know hearing previous speakers how important some of that license money is to your funding for your research. Is that ever a concern, or is that something you've seen in the past or anticipate seeing in the future?
Yeah, I don't, it's not a concern that I have overwhelmingly, particularly regulations on a single species. What I have seen more prevalent is frustration over regulations tends to make people less willing to participate in sampling. Like less willing to talk to an MRIP sampler, less willing to do the interview, less willing to put a tag and, report the tag. Even situations where, less willing to handle a fish nicely and return it to the water. I've seen situations where people are angry about regulations. They got to throw a fish back. They kill it out of spite when they throw it back. Or they're like, I'll let them things lay on the beach and throw them back, in 20 minutes when I'm ready. So I feel like that's had more of the impact on fishermen than not buying regulations. Not buying permits, but, I don't know, maybe someone from the division has seen anything like that. It'd probably be hard to track, though, because probably offset by increasing population, just all of our coastal areas.
Exactly. I think just recent years, a lot of what I hear is a recreational guy is, I'm not buying my license this year if I can't flounder fish. And then I guess another thing that can happen is those flounder fishermen focus on another fish. Maybe that fishery gets an unexpected hit, but I guess it's all kind of hard to track.
We call that the squeezing of the balloon. Particularly in the snapper grouper with so many species, and anywhere you have multiple things people can fish for, if you squeeze down on one fish, the balloon pops out of your hand in other directions, which means you end up having higher harvest on some other species. That's absolutely real because people want to fish. That's one of the things you have to realize. People want to fish. They want access to the resource. Increasing demands all the time. People are moving to the coast. My God, the number of boats that are around here. I say I've driven this coast from Key West to Portland, Maine. And the one thing that's common, the entire Atlantic coast, is everybody wants to live east of 95, and they want to be close to the water, and they want to have a boat, and a lot of them want to go fishing. And that's just continuing to be a demand, and it's hard on our environment, it's hard on our ecosystem, and it's hard on our resource.
Thank you solved all the problems.
All right, well, sure. We always do, don't we, Steve? Thank you.
Thank you, John. All right, well, that concludes all the talks we have scheduled today. I'm going to ask Ann Marquith and Holly White to make their way up here. They're going to provide some closing remarks. But before they get up here, I just wanted to take this opportunity to thank all the staff that contributed to the symposium as far as planning and setup and being here and just being available to interact with everyone that came and share their information. Ann and Holly are our two flounder leads for the division, and they took on a huge responsibility with planning this symposium. But there are also many other staff within the division that contributed to having this, be a success. Michelle Brodeur at the back, many of our other species leads, many of our, coastal habitat, our coastal programs staff, and just all of our administrative staff in the building. This was a collective effort by many staff, and I want to take this opportunity to thank everyone. And then Ann and Holly, leave us with some words of wisdom.
I don't know if we got words of wisdom. All right. Hi, everyone. For those who don't know me, I'm Ann Marquith. Thank you, Steve. And Steve kind of took the words out of my mouth. One of the first things I wanted to do was thank everybody who helped put this event together. It was as much as I hate this phrase, it was a giant lift for the division, and Holly and I, it's not necessarily something in our wheelhouse to put together a symposium, so anybody who helped with this, we thank you so, so very much. Also, thank y'all for sticking around today. It was a great day, and we had some really interesting talks. I've been the Southern Flounder co-lead for the last 6 years, and I unfortunately know how contentious the species can get. And today, it was just nice to take a step back from the management and just listen to everybody and learn some information.
So whether you have worked at a university and are directly helping to answer the research questions that the division has, you participate in the fishery and you allow us to sample your catches, whether you're commercial or recreational, you provide funding for the research that's ongoing, or just work at the division and help us collect the data we need for this species. You are part of a collaborative effort to help increase our knowledge, and we cannot thank you enough for everything y'all have done. There are two things I've gotten out of today, and one, that there is a lot of interest in this species, and there is a lot of effort being made to make sure we have the best information and data available so that we can manage this species properly, but there are also still a lot of questions that need to be answered. So please take what you have learned from today, relay it to your friends, take it back to your organizations, and just help us to keep collecting this information that is so important for this species. So thank you again for being here, and Holly?
Thanks, Ann. My name is Holly White. I am the Flounder co-lead. I just wanted to thank you all again for being here. Southern Flounder is one of the many special species that are found in North Carolina, which we've learned about today. And it's also one of the things that makes North Carolina really special and unique as well. So we just want to thank, again, to everybody, echo what everybody has said. Thank all the researchers and staff and everyone that stayed for joining us here today. I want to give a special shout out to Michelle and Debbie, if they're here, for all their help in facilitating this event, and as well as our Southern Flounder small group, and Steve and Kat for helping us get all this stuff going and encouraging our efforts here, and also to Mike for encouraging us to keep moving forward with this event. We hope that you'll leave here today knowing just a little bit more about Southern Flounder. And we as a division, and me personally as someone that's passionate not only about the resources, but as everyone as stakeholders, hope that you found the information today beneficial. As many have already echoed, our work here is not done, and today has shown that we're left with many new paths to investigate. So encourage to everyone to keep asking new questions. And with that, I'm just going to give a plug, two plugs actually. The first one is if you want to get more involved on the management side of things, we're always looking for advisory committee members to help advise the MFC or sit on our northern, southern, central? No. Finfish. Yeah, finfish, crustacean, all different types of advisory committees. So if you want to see Jesse Bissett here in the back, we didn't get to make it big enough there, but there's his email address, and we'll send out the reminder in our thank you email as well. But we would encourage anybody that would like to participate to please get up with Jesse and see how you can get involved. And the last thing is this QR code that is on the screen right now. That is for our exit survey. We'll also be sending everyone that has provided their email a link to the exit survey as well. So please, please, please fill that out. That'll help guide us, know how we did. Hopefully you guys have had a blast, and thanks again for joining us.